and get started. I didn't bother getting the screen up here because I thought it was more fun to show you that cover. I know you don't have a lot of free time for fun reading, but Mary Roach is an amazing nonfiction author, does a tremendous amount of, it's a wonderful combination of both scientific information in a very easily accessible format. She's done several really great anatomy and physiology books. Gulp was the one that was most appropriate for today's topics. But she's also done Stiff, which is a discussion of death and cadavers and corpses and things along those lines. She's done Boink, which is a great book about reproductive research, the reproductive system and the taboos that even today are still associated with that. Gulp, of course, on the digestive system. She's got a new book that just came out recently. It's not really anatomy and physiology, called Fuzz, about the interaction between animals and the legal system. She, like I said, she's an amazing, accessible, very engaging author. And so when you have some free time during the break, maybe with Christmas along the lines, you might want to look at some of those Mary Roach books. So that was the theme for today's quiz. Again, today we basically in lecture today, what the heck day is it? It is Tuesday, October 19th. Of course, 2021. In lecture and lab, we are going to finish up the rest of the digestive system. Anatomy, histology, function, all the things we've been talking about for that, because on Thursday, you have your big exam. As a reminder, because of the 5-day weekend, Tuesday when we come back, you have 2 pre-labs. Again, the pre-labs, not the reviews. The first one, not labs. The first one is Unit 22, and that's pages 580 through 584. And the other one is Unit 25. And that's pages 664 through 668. So not a tremendously long assignment, but something to just get you thinking about the respiratory system and the urinary system, which I know on paper don't seem like it would be necessarily very related, but as we'll see, there actually are some really key important relations between the respiratory system and the urinary system, which warrant them being discussed together. All right, so that is the game plan. Any questions on that? All right, excellent.
When were those pre-labs due?
They're due, oh, I'm sorry, you're right. I have the wrong date there, don't I? They are due, hold on, get rid of that. Tuesday, the 26th. I put 16 there instead of 26 by mistake. Excellent. All right, thank you for catching that. So yeah, just Tuesday when we come back, which is the 26th. Perfect. All right, excellent. Great question. Thank you for catching that. Any other questions before we dive back into the material? All right, we left off last class and we had just started discussing the liver and gallbladder, some of the accessory structures for the small intestine. We had already talked about the pancreas, so let's talk a little bit about the liver. We could really spend the rest of the semester talking about the things that the liver does. It is truly a rock star. It is the largest visceral organ, and it is comprised of, as we see here from the anterior view, not the 2 that are obvious, but actually 4 large lobes. Here, from an anterior view, we can really clearly see this nice large connective tissue structure known as the falciform ligament that separates it into the largest lobe, which is the right lobe, and the left lobe. Notice continuous with the falciform ligament is this ligament that sticks out, known as the Ligamentum Teres, or what is also known as the round ligament. Any idea what that round ligament might be? No one's looked ahead, no one knows? It's basically your internal belly button. What's your belly button?
Umbilical cord?
Right, absolutely. You've got the umbilical cord. What is the function of the umbilical cord?
To provide nutrients to the baby from the placenta?
Exactly. So from the placenta to the umbilical cord and your umbilicus region, oxygen and nutrients is carried in, waste is carried out. But obviously those blood vessels in the umbilical cord have to connect to blood vessels inside the abdominal pelvic cavity. So when baby's born, right, you take the scissors, you bite it, you rip it, whatever it is you do to that umbilical cord, and it shrivels up and becomes the belly button on the outside. Well, not surprisingly, the same way that the blood vessels from your digestive system go to the liver first, the blood vessels from mom go to the liver first. Because, right, again, the oxygen and nutrients that, or really the nutrients that they're receiving from the baby may be nutrient rich, but may not always be nutrient appropriate. So it needs to be processed by the liver first. So that that umbilical vein coming back to the heart actually goes to the liver first. And so when the umbilical cord is cut, that shrivels up and becomes the Ligamentum Teres. Notice we see the second accessory structure sticking his little head out here, the gallbladder, which is mostly posterior and inferior. So we can see it sticking its head out from the back here. And then of course, if we flip the liver around, there's a couple key things we can see. We just finished talking about the Ligamentum Teres, which we can see extending out. And here we get a better view of the gallbladder and its cystic duct, which connected to the ductwork of the liver. But we see two other important things as well. For starters, we see the third and fourth lobes. These, as you can see, are much smaller lobes. This is the caudate lobe located right here. And this small one down here is the quadrate lobe. And the way I remember them is they're always in alphabetical order, C first and Q second. But there's another interesting feature that can be seen here on the liver, and that is basically this indentation in the right lobe that we can see here from the posterior view. We can see that the right lobe actually wraps around the inferior vena cava. So the inferior vena cava, as we know, is located in the posterior wall of the abdominal pelvic cavity, and the liver basically wraps around it. And then, of course, we can also see, this may be the easiest way to identify them, some openings right here where those hepatic veins would come out. We can't really see it on this side, but there are some on this side as well that are coming into and feed into that inferior vena cava. So remember, it is the hepatic vein that leaves the liver to bring the blood back to the inferior vena cava, and then it is the hepatic portal vein that brings the blood from the digestive system to the liver. And we'll take a look at that and see that a little bit more clearly in just a minute. Actually, you know what? Let's do this first. I didn't think to get it set up ahead of time, so hopefully it will be there. That's a lame one. Let's see if I have it. Let's see how clever I am. All right, I'm clever. Excellent. Here's this great picture we can see of the liver model that's in the classroom. If you haven't had a chance to get to Open Lab, this is a model that's in the classroom. It's an excellent model. Notice this is an inferior view of the liver. We can tell that primarily one of the easiest ways to distinguish that as we look at this is we can see here's that notch of the inferior vena cava. So clearly, as we know, that's in the posterior part, and that's that notch in the right lobe. So this would be a person who would be laying down, face down. So they would be in a prone position, and their feet would be, their head would be forward and their feet would be behind us. So basically, if I tilted forward, this would be my liver. We can see that inferior gallbladder. Clearly, we can see the right lobe. Clearly, we can see the left lobe. And here we can also see the caudate and the quadrate lobes here as well. But the reason I wanted to show you this is because there are basically, as we expect, blood vessels that feed into and out of the liver. Here, as I just mentioned, coming out of the liver and into the inferior vena cava would be what blood vessel?
Hepatic vein.
Hepatic vein, excellent. So then what would this red blood vessel be?
Is that the hepatic artery?
The hepatic artery, excellent. And so what does that make this whitish? It's actually kind of more of a really light blue, but this particular, I guess the flash or whatever bleached it out a little bit. So this whitish looking blood vessel, what do you think this might be?
Hepatic portal vein.
Exactly. And let's think about this for a second. The hepatic portal vein, what is the condition of the blood in that hepatic portal vein?
Nutrient rich.
And?
Professor, you're not typing anything.
I am, it's just white. So you'll see it as soon as I let go. Here, we can do a light color like that and it'll stand out just as well. Nutrient rich, and how else would you describe it? Well, it's a vein. What do we typically know about systemic veins? There we go, excellent. I'm sorry, Ariel, I didn't see that you wrote there. Excellent. Nutrient rich and oxygen poor, spectacular. How does that compare to the hepatic artery? There we go, excellent. All right, its job is to bring oxygen to locations like the liver here to be processed. And of course, what is the liver processing? Well, blood coming from the hepatic portal vein. And once that processing is done, the blood leaves via hepatic vein. And what would you say the condition of the blood in that hepatic vein, or veins really, plural, should be?
Nutrient appropriate and
Excellent.
oxygen poor.
Yeah, absolutely. We've processed the blood, so it is now nutrient appropriate. However, that processing took energy. We need oxygen to make that energy. So once again, it is oxygen poor. So we actually have 3 blood supplies associated with the liver, 2 coming in, 1 oxygen rich, 1 nutrient rich. Then once it's processed, they're combined, they're processed, the oxygen is used, and then we have nutrient appropriate oxygen poor blood that then leaves the liver. All right, so most organs have just an artery that comes in, a vein that comes out. It's pretty simple, but as we know, there's a lot more going on inside the liver. And so we have these different blood supplies. And we see that when we look at the microscopic anatomy and of the liver as well. The liver, like I said, largest visceral organ. It's divided into 500,000 different liver lobules. These liver lobules are hexagon shaped. And these are basically the functional units of the liver. These hexagon-shaped structures are comprised of cells called hepatocytes. They are the workhorses of the liver. Their job, as we just finished mentioning, is to regulate the nutrient levels in the blood. But, right, any kind of toxins that are ingested with that food, with that drink, or somehow else gets into the body, they need to be detoxified as well. And if that wasn't enough, these hepatocytes, in their spare time, make bile. Someone remember, I think we talked about this in the last class, but what is it that bile does?
Emulsifies lipids.
Exactly. And what kind of digestion is that?
Mechanical.
Exactly. It is mechanical digestion. Basically, emulsification is taking that big chunk of fat and breaking it up into much smaller clumps of fat. We're not breaking bonds. We're not breaking that triglyceride into fatty acids and alcohols. Basically, what emulsification does by taking one large thing and breaking it down into a bunch of small things, it increases the surface area. And the more surface area it has, the more space there is for the enzymes to get in there and actually do the chemical breakdown that we want to occur. So the same way hydrochloric acid only mechanically digests protein, bile only mechanically digests lipids. Now, the hepatocytes are not the only cells found in here. We also have Coofer cells. Coofer cells are a special type of phagocyte. Normally, we think of phagocytes as being circulating cells, but these Coofer cells are actually anchored in the blood vessels of the liver, in the capillaries of the liver. They stay in place. Their job is to store lipid-soluble minerals, some heavy metals, things along those lines, but being phagocytes, any harmful pathogen that gets in there, it can gobble it up and it can present those antigens on their what class major histocompatibility complex?
2.
Class 2, which would activate what type of T cells?
CD4, helper T cells.
Which would become helper T cells. Excellent, spectacular. Again, which if you think about it, makes sense. We want our guards in the places where they're most likely to come in contact with stuff. And so much blood is coming to the liver, it totally makes sense to have these cells here. All right, let's look at the pretty picture of this. And as I said, those Coofer cells also store iron, lipids, heavy metals, some lipid-soluble minerals, vitamins, I'm saying, things along those lines. Here's the pretty picture from your textbook, but then let's go ahead and draw this so we can see this and the parts that make it up. So let's go to our whiteboard and let's do a really poor job of drawing this. So, oops. There we go. So there, and there, and there. So as I mentioned, these have a hexagon shaped shape to them. I know this isn't, I'm not going to bother fixing it. And at the center, right here in the center, and actually let's change the color of that. Right here in the center is a blood vessel. And this blood vessel in the center is what is known as the central vein. This central vein ultimately connects to our hepatic vein. Or we could say feeds into. Let's say that, I like that. So this is our central vein. It's going to feed into a hepatic vein. So based on that, as I mentioned, what do we expect the condition of the blood to be in this central vein?
Oxygen poor.
Oxygen poor and
Nutrient rich.
nutrient rich.
Oh, nutrient poor, sorry.
Nutrient appropriate, absolutely. It's been processed. This is where the blood ends up after it has been processed. As I mentioned, how am I going to do this easy? We'll just do it this way. There are these hepatocytes that are basically lined up in rows. And they form an array basically coming from all the sides towards the center of our liver lobule. I'll just draw one quick row on each of the sides. I won't bother putting the segmentation in there. But we have all these rows of hepatocytes that are lined up. And notice when I'm drawing these, there are spaces in between these rows. The reason there are spaces in between these rows is because between the rows in these spaces are a very specialized type of capillary. A capillary that has a very, very loose, incomplete epithelium to them, allowing for very large materials to get in and to get out. And what would we call those types of capillaries?
Sigmoid.
Close. It starts with an S. So it's not continuous, it's not fenestrated. So
Sinusoid.
yeah, there you go. Excellent. So we have these sinusoid capillaries. And these sinusoid capillaries are basically where the exchange of material takes place. So again, if there's too much glucose, that can be stored. If there's not enough magnesium, magnesium can be taken out of storage and it can be put into the blood. This is where the processing takes place. As I mentioned, this is also where you'll find those Coofer cells. So let's throw some purple Coofer cells in here that are checking out the condition of the blood as it is going through there. And then out at the periphery is where the blood is going to enter. Out here at the periphery, at all of the corners of our lobule is where we find what is known as the portal triad. And of course, with a name like a portal triad, how many things must it be?
3.
3, absolutely. And that portal triad includes a branch of, these are all, I'm going to write just hepatic artery, but it's a branch of the hepatic artery. And what again is the hepatic artery bringing to the party?
Oxygen-rich blood.
Yeah, it's bringing oxygen. And a branch of the hepatic portal vein. And what is it bringing to the party?
Nutrients.
Yeah, it's the nutrient-rich blood from the, and again, let's even be more specific. This is bringing nutrients from the digestive system, right? This is bringing the hepatic arteries, bringing oxygen from the lungs, right? Via the arterial system. So we've got those coming to the party. So at all of the corners, we are going to have a branch of the hepatic artery and a branch of the hepatic vein. Come on, there we go. So, as I mentioned, our hepatic artery feeds oxygen-rich blood into these sinusoid capillaries. Our hepatic portal vein feeds the nutrient-rich blood into these arteries and then into these sinusoid capillaries, and then it's processed, enters into the central vein, and then is released. So like I said, here the hepatocytes do the magic. This is where the processing of the blood takes place. So 2 blood supplies enter into the liver, one bringing oxygen, one bringing nutrients. They get processed by the hepatocytes, and then that processed blood feeds into the central vein, which feeds into the hepatic vein, which feeds into the inferior vena cava and leaves. Excellent. Questions on that? So that is the anatomy of the liver. Any questions? Really? None of you have a question?
The veins that are surrounding, I'm just confused on which ones are is that all hepatic artery branches?
So yes, again, these are, so we can put here. So these are all branches of, again, when the hepatic artery enters into the liver, it branches, basically feeding smaller and smaller blood vessels that are going to feed all of the hepatic arteries. So yes, in this case here, around this one liver lobule, you happen to have 6 branches of the hepatic artery and 6 branches of the hepatic portal. And again, remember, all these lobules are side to side. So there is going to be another lobule here that these will be in the corner of, and another lobule here that these will be in the corner of, and another one here. So again, we're going to have these arteries, these hepatic arteries and hepatic portal veins are going to be at the corners of many lobules. So excellent question. Yes, these are branches of those 2 blood vessels. Any other question?
You said 3 things.
Yeah, I said it was a portal triad. That was the 3 things. And how many things do we have in all the corners?
Only 2.
Only 2. So we need a 3rd thing. And that comes down to the hobbies of our hepatocytes. As I mentioned, as busy as the hepatocytes are, they actually have a little free time for their hobby. And what did I say their hobby was again?
Make bile?
Make bile, exactly. And so that's the other thing these hepatocytes are going to do. These hepatocytes make bile. And this bile needs to then leave the liver, where it's either going to enter into the small intestine, the duodenum, or go to the gallbladder. And so while we make that bile, it can't stay here inside of the liver. So what's going to happen is these tiny little bile canals, known as bile canaliculi, are going to collect that bile. Going to collect in those bile canaliculi, and they're going to feed into a branch of a bile duct. And so these are going to feed down and ultimately feed into a bile duct. And that branch of the bile duct is the 3rd component that can be seen at the corner of these hepatocytes. And so notice one other thing as well. What direction does blood flow? Towards the center or away from the center of our lobule?
Towards the center.
Yeah, blood goes towards the center. And which way does bile go?
Away.
Yeah, bile goes away. Again, I've done a truly amazing job of drawing this, but let's go back and look at the pretty picture from your textbook. Notice here, you can see all of these components that we have just finished talking about. We can see that central vein, which ultimately leads into the hepatic vein. At the corner, we can see our portal triad, made up of a branch of the hepatic artery, a branch of the hepatic portal vein, and a branch of the bile duct. We can see that the hepatic portal vein and hepatic artery are feeding into our sinusoid capillaries that are surrounded by the hepatocytes. And in fact, we can even see a Coofer cell that they have thrown in there, that hepatic macrophage called the Coofer cell, because good old Bob Coofer was the first person who identified them. And we can also see the little bile canaliculi that are collecting the bile, feeding it out into the bile duct. There's one other place where you can see this really, really nicely. Well, two places. One of those two places happened to be in the classroom. Where in the classroom we have this model. Notice this model. Oh, there it is. Okay, perfect. So here it is. So we've taken that three-dimensional structure here. Let's cheat and do it this way. What they have done with this particular model is basically they've taken a chunk out of the blood vessel, out of the liver lobule like this. So they've taken out a single slice. And that is what we're seeing here, a single slice of a liver lobule. Now, is this the outside down here, or is this, wait, there? Is this, why is that not writing here? There we go. Down here at the bottom, is this the outside or the inside of the liver lobule?
Outside?
Yeah, this is the outside. How do you know?
The triad?
Triad, excellent. So, and again, sure enough, we have three things. What's the red thing?
Hepatic artery?
What's the light blue thing?
Hepatic vein?
Nope.
Just kidding.
You're close though. What'd you forget?
Bile duct?
No, help her out. It's not the hepatic vein. What is it?
Hepatic portal vein.
Hepatic portal vein. Remember, hepatic vein and hepatic portal vein are not the same thing. And I much prefer you make that mistake here while we're discussing it than on the exam. So get used to that. That is the hepatic portal vein. And then, of course, the third thing, the green thing. What do you think that green thing is?
Bile duct?
Hey, what's that yellow thing? They got four things in their triad. What the heck is that?
Nerve?
Yeah, it's a nerve. Again, there's really no reason for it to be there. Nerves can play a role in affecting the liver, but it's not part of the triad. So again, one of those a little bit of artistic licenses. Notice our hepatic portal vein, our hepatic arteries feed into these squiggly things, these light blue things in here. What might they be?
Sinusoids.
Sinusoid capillaries. Notice these sinusoid capillaries are surrounded by these pentagon-shaped cells. What might those be?
Hepatocytes.
Hepatocytes. And those hepatocytes have this green mesh network around them. What might that be? Bile canaliculi. Excellent, Danielle. Excellent. Notice we know that bile is going to go this way from the hepatocytes out to the bile duct, whereas blood goes this way. So if we scroll to the inside, lo and behold, there's a nice big blood vessel at the center of our lobule. And what would that be?
Central vein.
Central vein. And what does it feed blood into?
Hepatic vein.
Hepatic vein. Excellent. See how easy that is? So I said there were two more places where we could see that. Where do you think the last one is? Where else can we see all of these features? Here's a nice illustration that shows it. Again, as we just talked about, we've hit all of that. Where's the final place you think we can see all of this anatomy?
Under a histology slide?
There you go, under histology slide. Excellent. We did all of that. So lo and behold, here we see a low magnification view of a liver. We can clearly see the liver lobules, the connective tissues that's dividing these into individual lobules. They may not always be perfectly hexagon shaped, depending on the plane of section, but they're always going to have a nice central vein in the center. And you can see all of the spaces between the hepatocytes where those big loose sinusoid capillaries are. The other thing that you can see is where the triad might be located inside of here. Now, we can't tell much by looking at it from this point of view. Here again, we see that nice central vein. We see those hepatocytes making up the spaces where those sinusoid capillaries are. But what I really want to show you is here at the center, on the edges, I should say, in between the individual lobules, you may have a structure that looks an awful lot like this. This is a hepatic portal triad. And conveniently enough, these three things happen to be numbered 1, 2, and 3. Based on everything you've learned up to this point in 431, what do you think number 2 is?
The one with the pink.
Yeah, what is that? Identify. So I've told you this is a hepatic portal triad. You know this is 3 things. And you'd get partial credit for that on the exam, Amy. If I asked you to identify this particular structure labeled 2 on the exam and you said artery, you'd get partial credit. But what do you need to say for full credit?
Hepatic artery.
Hepatic artery, absolutely. So that is our hepatic artery. How do you know?
That one looks like it has the most muscle tissue around it.
There you go, absolutely. Clearly it is lined. Oops, I should probably not use the, let's use the highlighter and make it tiny and make it green. A thin layer of simple squamous cells lining the inner surface. But the dead giveaway, as you mentioned, is a thick muscular wall. What does that make 1?
The hepatic portal vein.
Hepatic portal vein. Again, notice it's lined with a simple squamous epithelial tissue. And what about it makes it super obvious that it is a vein?
The largest lumen out of all 3.
Yeah, massive lumen. We know veins have massive, massive lumens. So it's going to make the vein super easy to be able to identify. So then what does that make 3?
And a bile duct.
That is indeed a bile duct. What makes you so certain it's a bile duct?
It looks like simple cuboidal epithelial tissue, which is makes up glands.
Exactly. Ducts, right, which are part of glands, absolutely exocrine glands. Ducts are typically made of simple cuboidal or simple columnar epithelial tissues. So the fact that it is a tube made up of simple cuboidal instead of simple squamous like we see in the blood vessels is a dead giveaway that it is indeed the hepatic portal, pardon me, the bile duct. Absolutely. Excellent. Notice as we look closely, there are some other structures here that could probably be identified. What of those three things do you think this is? Do you think that's an artery, a vein, or a duct?
Is that a bile caniculi?
Not a bad guess. Absolutely. I could see where you'd be tricked by it a little bit. This is actually another artery. Notice the giveaway is we can see the little bit of smooth muscle. Remember, it's lined by simple squamous, and sometimes with those simple squamouses, the squamous can be so large that, remember, the nucleus can bulge out. And so that's what you're actually seeing here. You're seeing the bulging. Notice this one had a little bit of bulging, not quite as obvious, but this is such a tiny artery that might even be an arteriole, but that small, something like that. But again, so we could see that. Whereas this, on the other hand, what is this likely?
Another bile duct?
Yeah, another bile duct, absolutely. Now, notice you and I are sitting here discussing this right now. This and this are not going to be on the exam. But will I have pictures like 1, 2, and 3 here on the exam and expect you to be able to distinguish these three components histologically? Yeah, absolutely. So I will have nice examples like this that can easily distinguish. So you should be able to easily identify the components of the hepatic portal, pardon me, of the portal triad histologically. All right, excellent. That is the gross anatomy and the microscopic anatomy of the liver. We still need to talk function. But before we talk function, I want to talk, we talked about this, I believe, at the end of the last class, but I want to briefly go over it again. And this is the duct system associated with the bile. As we mentioned, all those bile ducts we just talked about from all those bile canaliculi are going to feed into these two ducts that are the right over here, because again, we always think in anatomical position. So this is the right, and the left hepatic ducts, right? Because it's left hepatic because it comes from the left side of the liver, right hepatic because it comes from the right side of the liver. So notice they feed together into a duct that carries all the bile from all of the liver. So it is the common hepatic. If you're not digesting fats right now, that bile will travel up the cystic duct into the gallbladder, where it will be stored. And remember, as it's stored, it will be dehydrated, it will be concentrated, making it more potent. The bile in your gallbladder can be as much as 10 times more potent, more concentrated than it was when it was first produced, making it a much better emulsifier. What's interesting about the cystic duct is once you have that fat, you have that piece of birthday cake, and it's sitting there inside of your small intestine, then our gallbladder will contract and that bile will travel out the cystic duct. The cystic duct is the only two-way duct in this system. Every other duct just carries bile one way. And so notice the bile from the liver and the bile from the gallbladder feed into what is just the bile duct, because it contains bile from both organs. And that leads into where it meets that primary or main accessory duct of the pancreas and is released again, as we talked about last time, through that hepatopancreatic sphincter into that big hollow hepatopancreatic ampulla and out the finger-like extension, the duodenal papilla, into the duodenum. And what hormone causes the relaxation of the sphincter and the release of these secretions?
Is it cholecystokinin?
Cholecystokinin, excellent. Notice the bile just stores and concentrates Bile doesn't make it. So as we talked about in the last class, if you have bad gallstones and your gallbladder needs to be removed, you can still eat fats. The hepatic ducts will enlarge slightly, so the liver is able to store a small amount of bile, but can it concentrate it the same way the gallbladder does?
No.
No. So while you can still eat fats and you can still emulsify and digest fats, you're not going to be nearly as efficient as you were before. So you may have to go from that 3-scoop ice cream sundae to a single scoop ice cream sundae, right? So you're going to need to modify your diet as a result of that, but you are still capable of producing bile, storing a little bit of bile, and therefore emulsifying fats. All right. And again, here's that picture we saw last time. And again, 3 components. We have the sphincter, which is the enlargement of the circular muscle. We have the internal enlargement, which is the ampulla, and then that finger-like extension where it expresses out. That is the duodenal papillae. And again, we talked about those things a little bit. Cholecystokinin, the hormone cholecystokinin is what releases the hepatopancreatic sphincter. All right, that is the anatomy. Like I said, we could spend the rest of the semester practically talking about the function of the liver. The liver has over 200 different things that it is capable of doing. The good news is you don't have to learn all of them, just about 163. So that's not too bad. No, the good news is we can take those 200 plus functions and basically divide them into 3 main categories. And that's what we're going to worry about in this class. These somewhat related 3 main categories are metabolic regulation, hematological regulation, and bile production. Metabolic regulation basically is about regulating the composition of the blood. Remember, as I mentioned, we get that nutrient-rich blood coming in, and we need to make the blood nutrient appropriate. And remember, that involves 2 processes. That means taking out excess, but also adding deficient. So if we, like I said, if you don't have enough manganese in your diet, then stored manganese can be released from your liver. However, if you continue to eat a diet that is lacking of manganese, eventually what's going to happen? Not a trick question. Okay, let's take this to your checkbook. If you have money in your bank account and yet you keep writing checks, what's eventually going to happen?
It's gonna run out.
Yeah, you're going to run out. Absolutely. You need to eventually eat enough where you can store some more. Absolutely. Right. And again, lots of the foods, if you're not washing your lettuce or tomatoes before you're eating them, right, or other toxins that can be on there from those. So again, we need to make it nutrient appropriate. This plays an important role in helping us to maintain our blood glucose levels. As we know, our liver is one of the major topics for both insulin and glucagon to help us to maintain those glucose levels. But we also have to have the appropriate amount of lipids and amino acids and all of those types of things as well. Right. So again, make it nutrient appropriate. And again, the goal of this is to provide those resources and to help in the removal of wastes, or let's say toxins. We could say wastes too, for the other cells, right? So again, if other cells, for instance, when your muscles are producing lactic acid, your liver helps to break down that lactic acid. Right, so we want to have that nutrients in there so they're getting the glucose they're needing, they're getting rid of their lactic acid, make the cells happy. And again, by toxins, we can mean nitrogen wastes like lactic acid, toxins that you ingest, both medicinal and recreational drugs, and things along those lines as well. Now, similar to this, oh, and again, we also have enzymes that can activate or even produce regulatory hormones for other parts of the body as well. Again, it's all about regulation. However, obviously related to this, because it deals with the blood, is also our hematological regulation. Obviously, we have massive blood going into and therefore out of the liver. So at any one time, we have a large blood supply in our liver. And that's going to play a huge role in helping to maintain not necessarily the cells, although obviously if the blood's happy, the cells are happy, but this is really about maintaining the blood. Or the overall health of the body. One way it does that is by having those Coofer cells that are phagocytes and antigen presenting cells. This obviously can help to activate and stimulate our immune response. While it is important to have the right composition, to have the right amount of sodium, to have the right amount of glucose and things like that in the blood, a concept we're going to talk about a lot in the next section is the osmolarity. And osmolarity is really literally just the count of the amount of stuff in the blood. Counting how many things are in the blood, not so much what those things are, but just having the right amount of things. One of the main ways we maintain the osmolarity of the blood is with plasma proteins. And our most common plasma protein is albumin. And it is actually produced by those hepatocytes. But remember, as we talked about, maintaining the condition of the blood is more than just stimulating an immune response. As we've talked about, once we've defeated that harmful pathogen, we need to get rid of those antibodies, right? Once we send a hormone to tell a cell to make a particular protein, we don't want it making that protein from now to the end of time. So eventually we need to start breaking down those hormones to stop that signal. Again, yes, toxins and drugs, recreational, but in this case, as drugs, this more has a lot to do with the efficacy of our medicinal drugs, right? Some drugs you take as a pill, some drugs you take as an injection. What determines that? Is that determined by how mean your doctor is? If they're a nice doctor, they give you a pill. If they're a mean doctor, they give you an injection.
No, certain medications have to go in certain places or else they won't be absorbed correctly into the body.
Right, exactly. If you, for instance, you had a medication that the liver was really, really effective at breaking down, would you want to drink a shot of that or take that as a pill?
No.
No, because where's the very first place that medicine's going to go?
The liver?
To the liver. Once it gets absorbed by your digestive system, it's going straight to the liver. And if the liver is going to break down 75% of that medicine, right, you'd have to swallow a huge horse pill to get the right amount in your body to have some effect. So what do you do instead? You inject it into the muscle where it gets into the blood supply and it goes to the heart. And of course, once it goes to the heart, it goes everywhere. Some of that everywhere is going to be the liver. And any that goes to liver is pretty much going to be broken down right away. But there's plenty of other parts of that medicine that's going to go to the other parts of the body and have the effect that it needs. So again, this ability to break it down is really good and important so these things don't stay in our blood forever. But it also affects both the dose that we take and how we take our medicines. All right. Excellent. The third component, as I mentioned, involves bile. With all the free time that these hepatocytes have, and I guess they don't sleep or something because I don't know how they have free time, but depending on the composition of your food, the number of times you eat, the volume of the food that you eat, on average, your hepatocytes produce about 1 liter of bile during the course of the day. And of course, when not presently being used by the digestive system, it gets stored and concentrated in the gallbladder. This production, remember, is primarily controlled by the hormone cholecystokinin, and we'll actually see the mechanism by which this works in just a second. Bile contains some water, some cholesterol, some ions, but the two most important things I want to talk about are the bile salts and the bilirubin. The bile salts, as we talked about, are what are responsible for emulsifying our fats. These are the ones that are, again, going to provide that mechanical digestion of the fats. Anyone know what bilirubin is?
Does it break down red blood cells?
Yeah, as we know, red blood cells only last for about 100 to 120 days. And so during the breakdown of the hemoglobin, when that heme group is broken down, one of the components from it is that bilirubin. So it's from the breakdown of red blood cells. When bilirubin is broken down in the digestive system, it basically becomes two things. It is broken down into a urobilin. This urobilin is actually reabsorbed by the digestive system. And when it's reabsorbed by the digestive system, it's a yellow pigmentation that is actually released by our kidney. And so it actually causes the yellow coloration of your urine. The other component of the breakdown of bilirubin is stercobilin. This is a brown pigment that stays in the digestive system and basically leads the coloration of your feces. So by breaking down your red blood cells, your pee and your poop get their color. Of course, if we're not able to break down this bilirubin, bilirubin can build up in our blood if there's a problem with your liver or something like that. And what do we call that condition?
Jaundice.
Jaundice, absolutely. So if you're drinking vodka for breakfast, lunch, and dinner for the past 17 months, like I've been because we've been quarantined, right? That can affect your liver function. And so that bilirubin can't be broken down properly, and you can start to get jaundice in color. I'm not familiar with the term bile acid. I mean, bile is actually somewhat alkaline. So
I'm
I'm not.
saying this because one of my pregnancies, during my pregnancies, I had overproduction of bile acids, and I was trying to see which one was that, because the symptoms I got from overproduction of bile acids. I'm trying to make connection. What can that? What does that have to do with emulsification of fats? Because I got like really itching arms and like in between my fingers and stuff. So I was trying to see what can that.
I'm not familiar with the term bile acid. My guess is that, and I'm not sure how that would affect I mean, well, okay, I do know how that would affect that. My guess is that maybe you're not producing as much or too much cholesterol. Cholesterol can lead to fatty acid production, and that can make it more acidic. So it may not be the salt, it may be the other components of it that make it more acidic, so you could be overproducing that. The reason it can be a concern, as I mentioned, because where I was going to go with this is in the other location where you tend to see a fair amount of jaundice. Where else do you see a fair amount of jaundice, aside from people like me drinking alcohol all the time?
Like Bruce.
Babies, there you go, babies. Recent studies show as many as 30% of babies show some level of jaundice when they're born. The reason for this is fetal blood has a massive amount of red blood cells in it, more than a normal adult or a human would be. And so as a result of that, at the time of birth, a large amount of those red blood cells die. And if the liver isn't fully functional yet, it may have trouble breaking that bilirubin down. Bilirubin in and of itself makes the skin yellowish and things like that we talked about. But the other major concern, especially for infants, is that bilirubin can affect the nervous system, in particular the development of the nervous system. And obviously at that early age is a time when the nervous system is very, very active in its development. As a mature adult, you're not actively developing the nervous system, but can excessive bilirubin still infect nervous system? Yes. So my problem that itchiness or tingling sensation might have been due to an increase in bilirubin as a result of that, because it has an ability to affect the nervous system.
Yeah, they kind of explained me so that overproduction of my bile acids can damage baby. So the baby had to be born earlier.
Oh.
Before the third.
Oh, so this happened while you were pregnant. I didn't catch that part of it.
Yes, during the pregnancy.
Like I said, not my area of expertise, and I haven't heard of that before.
Yeah,
I can kind of tease out the pieces of it, but yeah. Well, hopefully you and the baby are both okay.
Yeah, sure.
Yeah, excellent. Well, then that's the most important part. And so that's probably the issue as well, is because with an increase in bilirubin, it does have the potential. I don't know if bilirubin can cross the placenta, but clearly if they were concerned about it, in high enough levels, it might. So that might have been why they were concerned. All right, because like I said, normally your bile should be a bit alkaline, a little bit basic, not acidic. So yeah, if some of the cholesterols or other fats or something like that make it more acidic, I could see where that would be an issue. We did that. And as we talked about, this, as we mentioned, increases the surface area. It doesn't break bonds. So again, this is part of the mechanical digestion of fats. What's interesting is that we release all these bile salts into our duodenum. And then the vast majority, over 90% of those bile salts, are actually reabsorbed with those fats. And we'll actually see that process, I think, after the break. And they can then, because if you think about it, if they're reabsorbed, where's the first place they go? Where does blood from the small intestine go?
To the liver?
To the liver. And in fact, a single bile salt, depending on the volume, the size, the composition of the meal, can be used as many as 5 or 6 times in the digestion of a single meal. All right, let's see how this process works. Here we have a pretty picture that shows this process, but I find it to be a teeny bit messy. So let's switch our view and go back to the whiteboard, and we can kind of do this in the whiteboard and a little bit more, less artistic and more schematic. All right, so this process begins when our chyme reaches the small intestine, the duodenum. All right. As we know, in the small intestine, we have enteroendocrine cells. That can produce a hormone based on the composition of the chyme. And when fats are present, what was the hormone that we said that was going to be produced?
Cholecystokinin.
Yeah, and again, for simplicity, I'm going to abbreviate this here, but don't do that there. Cholecystokinin. So when we talk about cholecystokinin, let's talk about its targets. One thing that it does is it targets the pancreas, and that pancreas is going to produce and release our enzymes that are going to help to break down the fats. In this case, that would be a pancreatic lipase. It targets the liver, and it targets the liver to produce and release bile. And it triggers the gallbladder to release bile. And lastly, it targets that hepatopancreatic sphincter to release the bile and the enzymes into the duodenum. All right, so far so good? That makes sense?
Yeah.
Okay, excellent. And again, we should be more specific. It releases that cholecystokinin into the blood. Bingo. And then it goes to these areas and tells them what to do. Oh, misspelled hepato. Excellent. This leads to the emulsification and digestion, so chemical and mechanical, down to the fats. And as I mentioned, and we'll talk about this process in a little bit more depth in a minute, but in general, this leads to the reabsorption, or let's say it this way, the absorption of the fats and the reabsorption of the bile salts. Now, when the fats get broken down, what vessel are they going to feed into? Oops.
Was that the lacteals?
Exactly. This is going to go into the lacteals. And we'll talk about what happens to that there. The bile salts, however, are going to go into the intestinal veins. And walk me through their mystical, magical journey. The intestinal veins feed into what? We're in the small intestine, so the intestinal veins. Good thing we didn't learn about the cardiovascular system, because then this would be cumulative and you'd be responsible for this information.
Superior mesenteric.
Which feeds into
Portal vein. Hepatic portal vein.
back to the liver. What's interesting is these bile salts stimulate the liver to make and release more bile salts. What does that sound an awful lot like?
Positive feedback.
Exactly. A positive feedback process where it produces and releases more bile to emulsify that fat even more, allowing us to absorb even more fats, which then brings more bile back, and the process continues until there's no fat left in the small intestine and homeostasis has been reestablished. And so, like I said, this is how some of these bile salts can be used 5-6 times in the digestion of a single meal. All right, and that's what the pretty picture from your textbook shows. Notice our enteroendocrine cells are releasing that hormone, the cholecystokinin, into our blood supply. Blood supply brings it to the, well, here, I guess it went the long way. Went the long way around. Clear. Went the long way around to the liver, stimulating the liver, stimulating the gallbladder to release the bile, right? Going to the pancreas to make the enzymes, going to the hepatic portal, hepatopancreatic sphincter, releasing that bile into the stomach. That bile salt is then reabsorbed, and as it reabsorbed, it goes into the blood supply, comes back and stimulates the liver to produce even more. So we get this positive feedback process of it going around and around until there's no more fats inside of our small intestine.
What was the definition of an enteroendocrine cell again?
Remember, they are single-celled endocrine glands that line the walls of our digestive system. Entero basically means within the wall of. So enteroendocrine, they're within the wall of the digestive system, just single-celled endocrine glands. Right? The G cells, they are enteroendocrine cells that we talked about in the stomach that produce gastrin. Right? Others produce others produce cholecystokinin, others produce secretin, others produce, gastric inhibitory peptidase, and so on and so forth.
Got it.
All right, excellent. All right, where are we? Perfect. We're a little late, but oh, actually no, because we had our quiz, so we haven't been talking too long. All right, so as I mentioned, there are going to be 3.5 pathways that I guarantee one will be an essay question on the exam. Again, we have that nice big cheeseburger for breakfast, and it needs to be broken down both chemically and mechanically. Know your enzymes. As I mentioned, there are also enzymes along the walls of the small intestine on those microvilli of the absorptive cells known as brush border. And these enterocytes are these cells on their microvilli have a large number of enzymes that help. Their job is basically to complete lactase, I should say, is a perfect example of one of these. You don't need to know the names of the individual brush border enzymes, just know them as brush border enzymes. So those you don't need to know. The enzymes you need to know are the ones that are released by the tongue, released by the salivary glands, released by the pancreas, released by the stomach. Right? You don't need to know the names of the specific enterocytes enzymes. Just collectively call them the brush border enzymes. We're going to break them down. Proteins get broken down into what?
Amino acids.
Excellent. Starches get broken down into what?
Glucose and maltose.
Yeah, simple sugars. I like that, simple sugars. Monosaccharides, absolutely. And then lipids get broken down into what?
Fatty acids.
Yeah, fatty acids and alcohols. Don't forget about the alcohols like glycerol and stuff like that. Absolutely. Once we break them down into those basic building blocks, then the vast majority, 90% of those building blocks are then absorbed by the small intestine. And like we talked about, most of it happens at the proximal end. And I left you with this picture at the end of the last class of this simple process of where this is going to take place. But after the break, when we come back, we'll put the pieces together and we will actually describe the 3.5 pathways that you absolutely positively need to know for the exam. All right. Questions on that? All right, let's go ahead and take our first break then. What time is it? It is right on the nose. 125. So that means coming back at 140. And I will start the recording at that point. All right, I will see you guys in 15 minutes. All righty. Now for the fun stuff. Okay, so we've talked about chemical and mechanical breakdown. We've talked about propulsion. Now our goal, again, is to get from the lumen of our digestive system into our body. And so technically to do that, we have to pass through two membranes. What are the two membranes that we actually have to pass through? I did turn the microphone on, right?
Yeah,
Okay, excellent. All right.
but it's a phospholipid bilayer, right?
Yeah, there you go. Absolutely. And not just one of them, but two, right? If you think about an absorptive cell is a simple columnar cell that have microvilli on the top of them. So if something is out here in the lumen, it has to pass through the top of the cell and the bottom of the cell. Of course, we're not going to get away with terms like top and bottom on an exam. So what would be the appropriate term for the top of the cell where the microvilli are?
Apical surface.
Yeah. The apical plasma membrane. And then that makes the bottom one what?
Basal.
Excellent. And are the mechanisms by which a molecule passes through the apical layer going to necessarily be the same that it uses to pass through the basal membrane?
No.
No. Luckily, way back in 430, we learned about membrane transport processes. All membrane transport processes can be broken down into two types. What are the two main types of membrane transport? Really three. What are the three main types of membrane transport?
Active.
Excellent. Active, and what else?
Passive.
Passive. Anyone remember the third?
Facilitate.
Say again.
Facilitated.
True, although facilitated is part of active and passive. Close though. It was vesicle, right? Where again, that would be endocytosis or exocytosis. Excellent. So let's make this a little smaller now. So again, as we mentioned, with a vesicle, if something is being brought in, what those two processes would that be?
Endo and exocytosis.
Well, coming in though would be which one?
Oh, endo.
Excellent. And if something was moved out of the cell, so let's say so this way, in to cell would be endocytosis, and out of the cell would be exactly exocytosis. Excellent. Perfect. Now, where it gets a teeny bit trickier is active and passive transport. Let's start with passive transport first. How do we define passive transport?
Not using any ATP.
Excellent. And how many different types of methods of active transport did we talk about in 430?
2.
Okay, what would they be?
It's like simple diffusion and then facilitated diffusion.
Excellent. Excellent. What does simple diffusion mean?
It can literally just pass right through. It doesn't need to have like an assistant.
Exactly. Passes straight through the plasma membrane without any problem at all. right? Whereas facilitated diffusion requires what?
A pump?
No, not a pump, because a pump would use ATP, but you got the right idea. We would need some type of protein that is going to facilitate, allows the substance in. And if you really remember back to 430, we talked about channels, which would be just holes in the wall, like your door frame is a channel that allows you into and out of the room, or like that revolving door at the hotel. And again, I don't mean the one that's plugged in and moving mechanically. I mean the one where you have to physically move it. And remember, we called those not channels, but we called those, does anybody remember? Where it undergoes a conformational change as it passes through. We called it carriers. Both of these don't use ATP. And of course, there is, I guess, a 2B. What is the other type of passive transport that we talked about way back in 430?
Osmosis?
Yeah. What is osmosis?
Diffusion of water?
Yeah, and really it's the simple diffusion of water. Water just passing through the membrane without really any assistance. So it kind of is part of simple diffusion. It's the simple diffusion of water, but it's important enough where we can call it its own process if we wanted to. Excellent. Active transport, what does that mean?
It uses ATP?
Although, can we be a little more specific in our definition? You're absolutely correct. That's 100% correct. But we can also be more specific in that definition as well. Anybody remember our more specific definition of active transport?
Directly uses ATP?
Does it always have to directly?
No.
Right, where either the, because again, inactive transport, it always uses a protein. So it is a facilitated transport, but those proteins are either going to directly or indirectly use ATP, right? Primary active transport is our pumps, like you mentioned, where we use ATP to move a substance across the membrane. So what was secondary active transport?
Does that use a G protein?
Not a bad guess. No, there are some that can use AGP GTP or GAP or a GMP, those kind of things. Now, remember with a secondary active transport, this is where the protein itself doesn't use ATP, but we need to indirectly. We had another fancy name for these secondary active transporters. We called them co-transporters. A co-transporter basically uses one molecule, like most commonly sodium. It uses the energy, let's say it that way. Right? Where is there a lot of sodium? Outside the cell or inside the cell?
Outside.
Outside the cell, right? What is the charge of the cell on the inside? Is it positive or is it negative?
Negative.
Negative. So notice it's negative inside the cell. So sodium being a positive ion really wants to come inside. There's more sodium outside than there is inside. So it wants to go down its concentration gradient. So remember, sodium really, really, really wants to enter into a cell. So we let it into the cell. We say, okay, sodium, you can come in, but you coming in, I'm going to use your energy to do some work, to either bring in something else or kick something else out. And in this case, it's going to be to bring something else in. And if you remember, we had a fancy term for that as well. We called those symporters. Now, remember, sodium only does work because it really wants to come inside. If we keep letting sodium come inside, the inside of the cell is going to get positive. If we keep letting sodium come inside, the concentration gradient is going to be lost. So I can get sodium to come in and do work for me, but as soon as that sodium gets inside, what do I have to do?
Kick out some other molecule.
Yeah, I need to use a pump to kick it out. Say, all right, fine, sodium, you're inside now, but now you got to get out. And of course, to get sodium out, what am I going to have to do?
Use sodium potassium pump.
Yeah, that's one way, but you've got the right idea. I'm going to have to use a pump, and pumps use ATP. So remember, in our case here, it is indirectly using ATP. I let sodium to come in to do the work. Sodium's bringing the substance in, but then I have to use ATP to kick the sodium out. So that's why it's an active transport, because it is using ATP. It's just using ATP indirectly. I love how many of you were able to respond to all of this and remember all of this. If you weren't as comfortable with this, then I strongly encourage you to go back to chapter 3 and review your membrane transport mechanisms and make sure you understand them. Now, with our digestive system, it is mostly going to use co-transportation and facilitated diffusion, but not all. And so we'll see those different mechanisms as we go through it. Lastly, because we have talked about the lymphatic system, and because we have talked about the cardiovascular system, and because this is a cumulative class, once we get that absorbed into the interstitial fluid, out of the lumen into the interstitial fluid, we already know the paths of where it's going to go. So on the exam, when you're describing this process, not only do you need to tell me how it gets into the interstitial fluid, but you have to give me the path to the right atrium. And luckily, we'll go through this together. All righty. With this information in mind, let's work through this. Your book does a great job of describing this process. Hopefully, you've already looked at it and thought about it that way. So I'm going to do it a little bit differently from the way your book describes it. Let's start first with our carbohydrates. All right. So I'm going to set mine up a little differently from the way our textbook does this. Let's go with the macromolecule we are going to break down, and then what it is broken down into. All right. We are going to figure out all the locations where the mechanical digestion takes place and what that mechanical digestion is. We will then identify all of the chemical digestion that takes place, plus the enzymes, where and the enzyme and where that enzyme is produced. We then, for simplicity, there's my absorptive cell. And then once it's absorbed, as I mentioned, we are going to need to know the pathway to the Right atrium. So this is the information we need to know. So like I said, here, we need to cross the apical surface, and we need to cross the basal surface. Excellent. All righty. So, like I said, we will start easy first, and with easy, we'll start with our carbohydrates. Oops, hopefully I spell that correctly. Carbohydrates are broken down into what again?
Simple sugars.
Simple sugars, which are made up of either 1 or 2 carbon rings. So what would those be called?
Monosaccharides.
Yeah, monosaccharides. Excellent. So broken down into monosaccharides. So let's think about, where does the mechanical digestion of carbohydrates begin?
In the oral cavity.
Okay. In the oral cavity with what process?
Mastication.
Awesome. Does it undergo any other mechanical digestion?
In the stomach with churning.
Excellent. Does it undergo any other mechanical digestion?
Segmentation in
There
the
you
small
go.
intestine.
Excellent. Excellent. Perfect. And actually, if I think about this, is this true just for carbohydrates?
No, doesn't all of them go through this?
Yeah, so pretty much aren't these going to all be true for all of our possible macromolecules? Yeah, so let's change the color of this to brown to remind us to leave this here, because we're not going to change it. That's going to be the same for all of them. Excellent. However, is the chemical digestion going to be the same for all of them?
No.
No. Excellent. So where does chemical digestion of our carbohydrates begin?
In the mouth.
Excellent. With what?
Salivary amylase.
Which is produced where? Oops. Salivary amylase. There we go.
The salivary glands.
Yeah. Excellent. Anywhere else?
Pancreatic amylase in the pancreas.
Oops. And where is that pancreatic amylase active?
Small intestine.
Yeah. Excellent. Yep. And let's not forget that there's also those brush border enzymes also in the small intestine as well. Basically, the salivary amylase and the pancreatic amylase break the complex carbohydrates, the polysaccharides, into monosaccharides and disaccharides. And then our brush border enzymes basically break it down into our monosaccharides. Excellent. So those are 3 mechanical digestion process, 3 chemical digestion processes. And so at the end of that, here in our lumen, we have our monosaccharides, which we need to get across our apical surface. Now, based on yours and mine and pretty much everyone, especially in the United States, what does our diet primarily consist of?
Cheeseburgers?
Okay, absolutely, cheeseburgers. And of course, then the fries that come with that, and the shakes that come with that, and the potato chips we had as a snack, and that big bowl of cereal that we had for dinner last night. And so notice we have a very carbohydrate-rich diet. As such, do you think we need to use a massive amount of energy to get as many of those sugars into our cells as possible? Remember, this is kind of a very key point in the digestive process. Right here, this apical lumen is kind of a key point, because remember, here in the lumen is outside the body. Anything that stays in the lumen stays outside of the body. Once it gets into the cell, then it's now inside the body. So getting across this first surface, this apical surface, is really, really important. So with a carbohydrate-rich diet, do you think we need to use a massive amount of energy to make sure we're getting enough glucose into our body? Are you getting enough sugars into your body on a daily basis?
Yes.
Yeah, probably. So not surprisingly, it is going to primarily enter via co-transportation and facilitated diffusion. So that is going to be the primary way we're going to get those simple sugars, those monosaccharides, into the cell. Now, once they're in the cell, they're safe. We're not going to lose them anymore. Do we really need to do anything to modify them or change them in any way? No, in fact, that kind of sounds like a silly question, doesn't it? It is, but we'll see why it's not later. But when it comes to getting these things out of the cell, if we have a very big glycogen-rich diet and we're absorbing lots of monosaccharides into the cell and we get a very high concentration of these monosaccharides in the cell, do you think we're going to require any energy to get them out of the cell, out the basal surface?
No.
No. As we know, right, diffusion goes from a high concentration to a low concentration. And since there's no risk of losing them anymore, we don't want to waste any energy here. So we are going to use just facilitated diffusion to get it out, the basal surface. And then from there, it's into the interstitial fluid. Questions on that? Excellent. Once in the interstitial fluid, there are one of two capillaries it can go into. What type of capillary is it going to enter?
Lymphatic?
So a carbohydrate's going to go into lymphatic capillaries?
Blood capillaries.
Blood capillaries. Excellent. It's going to go into the blood supply. Remember, it's primarily the lipids that are going to go into the lymphatic, the lacteals. Excellent. So now let's figure out how we get back to the heart. That blood vessel capillary feeds into what?
Intestinal vein.
which feeds into
The superior mesenteric vein.
which feeds into
The hepatic portal vein.
which feeds into
Sinusoid capillaries.
which feeds into
Central vein.
excellent, which feeds into
The hepatic vein.
which feeds into
The vena cava, inferior vena cava.
which feeds into
The right atrium.
see how easy that was? And there you go. Just that simply, using all the information you know, we have now traced the path of our carbohydrates from the bread of that cheeseburger you had for breakfast all the way to the point where that carbohydrate, that monosaccharide, gets to the right atrium of your heart. And from the right atrium, the heart, where is it going to go?
Into
Trick
the
question.
body.
Yeah, absolutely. Well, technically it goes to the liver first. I mean, to the lungs first. But once it comes back from the lungs, you're absolutely right. It can be distributed throughout the entire body. Absolutely. Right. Once you get back to the heart, you can be delivered anywhere. And there you go. Just that simply, we have described this process in carbohydrates. Notice your book does a good job of describing this process as well. It's got this nice flow chart. Notice again, just like we talked about, it starts with that carbohydrate in the oral cavity where salivary amylase starts the chemical breakdown and our mechanical breakdown occurs. So that we have tri and diglycerides by the time we get into our stomach, where we have that churning that is going to take place. Where we get to the small intestine, where our pancreatic amylase continues the breakdown of those sugars. And then finally, our brush border enzymes like maltase and sucrase and lactase break down those diglycerides. And again, you don't need to know those names. Lactase is an easy one. What does lactase break down?
Lactose
Yeah, where do you get lactose?
from milk.
Yeah, exactly. Breaks them down into our simple sugars, our monosaccharides. From there, they are going to cross the apical border primarily by facilitated diffusion and co-transport to get those monosaccharides into the cell. Once it's out of the lumen and into the cell, there's no risk of losing it anymore. So there's no point in wasting any energy getting it out of the cell, facilitated diffusion on the basal surface. And as we know, it then enters into the interstitial fluid, goes into a blood vessel capillary, and is transported to the heart via the liver. All right. And one more pretty picture where, again, it does a good job of showing where this breakdown is occurring, the enzymes and stuff involved of breaking down that sugar, that starch in that potato or in that bread that you had for breakfast into the monosaccharides that you absorb into your body. All right. Questions on this?
Are all the pathways to the right atrium the same?
Let's find out. That is an excellent question, which is why we're going to go through all of them. So let's cheat right now. We are going to go ahead and do, next, we're going to do our proteins. And proteins, let's go ahead and change colors. Proteins, of course, because it starts with P, have to be purple, are broken down into what?
Peptides and amino acids.
Excellent. Now, let's figure this out. Based on what you know, let's talk about the end of the story. Once it exits the cell off the basal surface and is in the interstitial fluid, where are those amino acids going to go?
The same place.
Yeah, they're going to enter into blood capillaries. And if they enter into the blood capillaries, what do you know about their pathways?
It'll be the same.
Exactly, it's going to be the same. So you are correct. For our carbohydrates, I know you're absolutely 100% correct. For our for our carbohydrates and for our proteins, the pathway is the same. However, there's also lipids. And being the smart, sophisticated students that you are, if the pathway was the same for lipids, would I be making you learn it?
No.
Probably not.
No. So they're not all going to be this way. There is going to be a difference, but you are absolutely correct. We've already spoiled the ending of this story because now we know it's going to end the exact same way as our carbohydrates did. And here, let's go ahead and make those red. Perfect. All righty. But let's work our way up to it. Proteins need to be broken down into amino acids. That, of course, begins in the mouth with mastication, continues in the stomach with churning, and is also continued in the small intestine with segmentation. Is there any additional mechanical breakdown of proteins that occur that didn't occur in our carbohydrates?
From hydrochloric acid
There you go. Where does that take place?
in the stomach with the parietal cells.
Excellent. In the stomach, our parietal cells produce hydrochloric acid. And what would we call that process? That what hydrochloric acid actually does to these proteins?
Denatures them, like natration.
Denature it, absolutely. It denatures the proteins. Again, you could also say that it breaks the hydrogen bonds, changing the shape. All right, which is, again, 100% true, but denaturing is a lot less letters to have to type. So either way that you want to describe that, absolutely, that's fine. So in the stomach, we denature the proteins, we change their shape by breaking those hydrogen bonds using that hydrochloric acid made by the parietal cells. So we do have a fourth unique mechanical digestion process for proteins. Excellent. Chemical digestion. Where does chemical digestion of our proteins begin?
In the stomach.
Via what?
Pepsin released by the chief cells.
Excellent. Anywhere else where the chemical digestion of proteins occurs?
You have trypsin and carboxytrypsin. Is that a word?
Carboxypeptidase. So there's chymotrypsin, trypsin, and carboxypeptidase. As long as you know the primary one, which is trypsin, I'll be okay with that. So we know trypsin. What makes the trypsin?
The pancreas.
but where is it used?
Small intestine.
So in the small intestine, we get that trypsin and the chymotrypsin and the carboxypeptidase. But as long as you know trypsin, I'll be fine with that in there. And then again, we can't forget our brush border enzymes, which again are in the small intestine. And those are the three ways that we chemically break down our proteins to end up with amino acids here in the lumen of our digestive system. Now, how important are proteins to the function of the cells of our body?
Very important.
Super important. Remember, if you think way back to 430, one of the things that we said is 20% of your body weight is proteins, which I know doesn't sound like a lot until I remind you that 60% of your body weight is water. So you're only 40% stuff, and half of that stuff is proteins. So it is super important to make sure we find every single one of these little amino acids in that cheeseburger you had for breakfast and get it into our cell. So we're going to use a massive amount of energy. And so we are going to primarily use both primary and secondary active transport. Or if you wanted, you could also say we will use pumps and co-transporters. I would accept that as well, because our pumps are our primary active transporters and our co-transporters are our secondary active transporters to get those things into the cell. Now, remember, we could be a little bit more lackadaisical about getting those carbohydrates out of the cells. But remember, all of the cells of our body really want these amino acids. So do we want them lollygagging inside of this cell for too long?
No.
No. So yeah, we don't want to waste energy. We've stopped it from being lost. So we will use some facilitated diffusion. But there's going to be a lot of other things that are going to want to get out of this cell as well. And we can use those to do work to get our amino acids out as well. So we can use our facilitated diffusion and we can use co-transporters. Primarily, again, there's always exceptions to all of these, but primarily those are the things we're going to use to get them out. And again, do we have to do anything to these amino acids before we can get them out of the cell? No, it's a silly question. So there's no change that is taking place with these amino acids. So they're just pumped right out into the interstitial fluid where they enter the blood capillary. And from there into the intestinal vein, into the superior mesenteric vein, into the hepatic portal vein, into the sinusoid capillary, central vein, hepatic vein, inferior vena cava, right atrium of the heart. And just that simply, we've done the same thing for proteins. Again, your book does a great job of describing this. It's got this nice flow chart. Again, starting in the stomach, we use our pepsin, right? Don't forget about the chomping and chewing that's taking place in that oral cavity, right? Churning in the stomach, segmentation in the small intestine, our pancreatic enzymes, our brush border enzymes to get those amino acids. And once we get those amino acids, we want these amino acids saved. So facilitated diffusion and co-transport to get them across, but also with active transport as well, primary as well. It doesn't say this here, but we are absolutely going to use primary as well. Active transport to get these things into the cell. And again, once it's into the cell, it's not as much of a concern, but we still want to make sure that we can get it to the cells. So we're not going to just rely on diffusion. We'll use some co-transport to get it out as well. Into the blood, and then from there, we're comfortable about where it goes. And again, your book's got a nice picture that talks about the enzymes and the processes and where it's all taking place. So great job of doing this and describing these things in your textbook. And for proteins, they even have a pretty picture. Notice here they show a co-transporter. Sodium comes in and brings that amino acid in with it, and then just facilitated diffusion out and into a capillary. All righty. Questions on proteins?
Yeah, I had a question.
Yes.
You said that when it goes into the apical surface, it doesn't change, correct? You said.
So yeah, it's an amino acid in the lumen, and it's an amino acid when it's inside,
okay.
and it's an amino acid when it enters into the interstitial fluid.
And does that go the same as carbohydrates as well?
Yeah.
So the macro.
Yeah.
Okay, I just wanted to make sure.
Yeah, but
right, thank you.
You've absolutely hit it on the head. It is a point I've emphasized, just like I've emphasized the pathways. So notice lipids are going to be different.
Okay, thank you.
Yep. All righty. So it's a great question, and I love that that's tickling you in the back of the brain. You're like, why is he making a point of emphasizing this? Because nothing's happening, and that doesn't make a lot of sense. But you've absolutely hit it on the head, because as we're going to see in this third pathway, things are a little bit different. All right. So great question. Any more before we finish up proteins? All right, then let's set our path. All right. Lipids. What are lipids broken down into again? What are their basic building blocks?
Alcohols,
Fatty
fatty acids.
Excellent. They're broken down to fatty acids and alcohols. Big three: mechanical digestion, mastication in the mouth, churning in the stomach, segmentation in the small intestine. Any unique mechanical digestion process that takes place for lipids?
Emulsification.
Exactly. And where does that occur?
Yes, small intestine.
Excellent. In the small intestine. Ow. What causes this emulsification?
The liver produces and releases bile salts, or the bile.
Yeah, we could say bile salts. It is the bile salts component. And we can't forget the gallbladder, since it doesn't make it, but it does store and concentrate it. So it doesn't hurt to put in there. I wouldn't mark you wrong if you forgot to put the gallbladder there, or didn't forget to, but if you didn't put the gallbladder there. But for teaching purposes, I'll throw it there now. But you've got a right idea. Bile salt. And again, if you'd said bile on the exam, I wouldn't have marked it wrong either. But let's be precise while we're going through this together. Bile salt from the liver, stored in the gallbladder, helps in that emulsification of the fat in our small intestine. Again, part of that mechanical digestion. Excellent. All right, where does chemical digestion begin?
In The mouth?
So again, I like where you're going. Why do you say mouth?
Well, isn't there lingual lipase in the mouth? Released in the mouth?
Yeah, it is released in the mouth, but like we talked about, if I took a little bit of fat and I put it on my tongue and I just let it sit there, would it be digested by that lingual lipase?
No, is it inactive?
It's inactive. So where does it become active?
In the small intestine?
Close. What causes it to be activated?
Is it a pH change in the stomach?
There we go.
Uric acid?
Again, so lipids are a little bit tricky. You absolutely have hit that all on the head. In our mouth, specifically our tongue, produces a lingual lipase, but it produces it in an inactive form. It's not till the pH change of reaching the stomach occurs that it starts that process of breaking it down. So again, it's produced in the mouth, but it doesn't become active till the stomach. Any other chemical digestion?
Pancreatic lipase.
Which is, of course, made from the pancreas. And of course, while it's made in the pancreas, where is it used?
Small intestine.
Excellent. So in the small intestine, we use that pancreatic lipase to break down the pancreas. And again, for consistency, we'll put brush border enzymes in the small intestine as well. And again, at this point, at the end of this, we then have our fatty acids and alcohols. And that's what we have here in the lumen. Our fatty acids and our alcohols. However, these fatty acids and alcohols, remember we talked about how we reabsorb the bile salts? They actually mix with the bile salts. And when they mix with the bile salts, as we talked about, emulsifies this fat. But then it also mixes with these alcohols and these fatty acids, and it forms these little clumps called micelles. These micelles, and since we're using blue, is a clump of fatty acids and alcohols mixed in with these bile salts. Now, fatty acids, alcohols, right? These things are lipid soluble. This micelle that is formed is a lipid soluble structure. So what does it need to do to cross the apical surface?
Diffuse?
Yeah, and not just any diffusion, but just simple diffusion. We can't stop it from coming in. Well, technically we can. How are we on time? We're doing good time-wise. All right. Back in ancient times, and by ancient times, I think I was like in high school or something like that, there was a special fat that had been found that had been given the name alai or something like that. Basically, it was a fat that our bodies couldn't digest and break down. And because it couldn't digest and break down, it couldn't be absorbed by your digestive system. Right? That's the problem. You eat fats, it's going to get into your body. It's simple diffusion, it's getting inside, and we all want to be beautiful on the outside, so we don't want fats getting in. So you could eat less food, or if they use this special fat to, say, cook potato chips, then when you ate the bag of potato chips, you wouldn't absorb any of the fats from that, and so much less fat would be absorbed into your body. which is awesome. Less fat gets into your body. You get to look prettier on the outside. What's the downside of this? Well, if the lipids aren't absorbed into your body, where do they stay?
In the plasma membrane?
Not in the plasma membrane, but inside the lumen of your digestive system. No, because remember, it doesn't get into the blood vessels. It doesn't get into the body. It stays in the lumen of the digestive system, right? You never absorb it. It never enters the body, which is awesome, except it has to then leave the lumen of the digestive system, right? You eat a whole bag of these chips and you have a massive amount of this fat inside of the lumen of your digestive system. And guess where that goes? Out the other end, right? So you would have these pools of oils in your defecation. You would have very oily and very explosive flatulence, right? And other kind of problems. There were pills that would do the same thing. And these pills would basically stop you from breaking down the fats in the food that you ate. And it's been such a long time since I looked at it. They didn't call it side effects. They had some quirky term for it. But like, for instance, when you first started using these pills, they recommended you only wear dark colored pants and not roam too far from your bathroom, and know that it was not uncommon to find pooling of oil or to have this explosive diarrhea or all these kind of things, right? All in the name of being beautiful on the outside. And shockingly, as you can tell, this isn't something that's taken on because you don't see any of this today because again, it didn't turn out to not be worth it. But yeah, normally it just gets in by simple diffusion. So when you eat fats, it gets inside. Now, something interesting happens once these fats get inside. This micelle breaks up. I may have to make this smaller now to fit this all in here. When the micelle breaks apart, it separates into the bile salts. And as we have already discussed earlier today, we know those bile salts are going to leave into the interstitial fluid. They are going to end up into the blood, and they're going to go back to the liver, right? In that positive feedback process we talked about. So we know what's happening to the bile salts, but as I mentioned, the micelle breaks up into bile salts, and it also breaks up back into those fatty acids and alcohols. And those fatty acids and alcohols are remade into lipids, like our triglycerides. Or our steroids, or other big complex lipids. Now, as we know, these big, huge, complex lipids, can we just release that into the interstitial fluid? Does
No.
water like fats?
No.
No, it'll congest and it'll congeal. So then what we have to do is we have to coat this big structure. These are bound together, and they're then coated with water-soluble proteins to make a structure we call a chylomicron. This is a big, huge structure. Big, huge mass of fats with a big, huge protein coat wrapped around the outside. Is this the kind of thing, do you think we can have a channel that can just allow this to leave the cell?
No.
No. So how are we going to get this out of the cell? If we have something really, really large, how can we get something really large out of the cell?
Exocytosis?
Exactly. The exocytosis. So that big, huge, large purple protein coated chunk of lipids. So there's my lipids. Coated in those proteins is going to enter into the interstitial fluid. And this big, huge chylomicron, is this going to be able to enter into even a fenestrated capillary, do you think, into our blood supply?
No,
So where does it enter?
the lacteal.
Yeah. Remember, those our lymphatic capillaries have those big swinging doors that is going to allow this material to enter into our lymphatic system. So it is going to enter into a lacteal, which is going to feed it into what?
Lymphatic collecting vessels.
I like that. Of course, these are going to be the afferent and efferent. And what determines whether something's afferent and efferent?
Whether they feed into or go out of a lymph node.
Excellent. Ultimately, these collecting vessels are going to feed into one big trunk. And what's the one big trunk it's going to feed into?
So the thoracic.
It's not the thoracic trunk. I think you're thinking duct. Which trunk is it going to feed into?
The intestinal trunk.
Which feeds into what?
The cisterna chili.
Excellent. Which feeds into what duct?
The thoracic duct.
Which feeds into
Left subclavian vein.
which feeds into
The left brachiocephalic vein.
which feeds into
Superior vena cava. And then the right atrium.
There we go. Isn't it fun when you get to use the information you've already used in a class again? It's not just, throw it in the mind, purge it, and never have to think about it again. It's fun when the things you use actually make sense and are useful. At least it is to me. So notice this one's a little bit more elaborate. And normally if we were sitting in the normal classroom, I know the warning bells will go off, because if you have three similar processes and one is harder than the others, and that's the one that's likely to be on the exam. But I will remind you that all of these plus one more we'll talk about is in a pool of test questions, which you'll randomly get one assigned to you. So I literally have no idea which of these essay questions you will get, but I know it's in there. What makes you say that high fat diets help you to lose weight?
Diets like keto diet, when they don't eat much, they don't eat carbohydrates, they eat little protein and high fat. And I've seen so many people losing.
Okay, I see where you're going with that. The point of the keto diet is to change the metabolism in our bodies. As we've talked about before, carbohydrates are very simple and easily to break down. And even proteins are very easy and simple to break down. Yes, adipose is stored energy, but it also provides protection, provides insulation, and things along those lines. So for instance, right, with summer right around the corner, I know it's not, we just finished it, but right at the beginning of summer, we were all trying to get that beautiful bikini body. So you go on the popcorn diet, where you eat one piece of popcorn for breakfast and one for lunch, and a reasonable 3 pieces of popcorn for dinner. Right? Clearly, if you were starving yourself that way, your caloric intake would be less than your caloric output. The problem with that is when your body goes into starvation mode, what happens is that it makes it easier to break down glucose and easier to actually break down proteins to get the fuel and the energy that you need. Fat is a valuable resource, and in starvation mode, your body actually tries to hold on to it. When you change the metabolism of your body with a keto diet, the goal is in entering ketosis, a state of ketosis, is that it makes the body more likely to break down lipids than carbohydrates to get the energy necessary for your fuel. So the goal with that is to try to convert the body into being more fat burning as opposed to carbohydrate burning. So by lessening the amount of carbohydrates, by lessening the amount of proteins, you can kind of get your body in a state where it's going to be more likely to break down lipids. So you can't just eat massive amounts of them, but you are right that the composition of your diet changes in such a way to try to essentially trick your body into more readily metabolizing lipids.
Yeah, that makes more sense.
Yep.
Thank you.
Yep. All right. Great question. Any others on this or this pathway?
Yeah, my dogs are barking, so I don't know if you said this, but the chylomicrons were too big to enter a blood vessel, so they enter a lacteal. Is that it?
Yes, and they're also too big to just leave the cell in a channel. So that's why they leave via exocytosis.
Okay.
Yeah. Your book, again, has some really nice pictures that show this. But again, there's one major issue that I have with it, and I'll point it out to you so that hopefully it will not confuse you. So again, here's the lingual lipase produced in the oral cavity, but remember, it doesn't become active till the stomach. We talked about the bile salts, pancreatic lipase, brush border enzymes, all of those things we've talked about. But again, we are now going to take those fatty acids, take those alcohols, the monoglycerides, things like that, and make that micelle. That micelle, being lipid soluble, just passes the apical surface via simple diffusion. Once inside, we take those fatty acids and those alcohols and remake proteins. Remake fats, lipids, like triglycerides, like phospholipids, like steroids. And we clump those together and coat them with a big, large protein coat to form that chylomicron. And again, anything that big can't just pass out a channel, so it has to leave via exocytosis. And anything that big isn't going to be able to get into the blood vessel. But luckily, with that swinging door of our lacteal, it can get into there. Again, they have this nice picture. They have this nice picture again, talking about the process. This is the picture that I have a little bit of a problem with. It's 100% correct, but their scale is a little bit off, right? Notice how massive the fat globule is. And then our emulsified fats are a little bit smaller. And then we have our micelles and then tiny chylomicrons. What's bigger, a micelle or a chylomicron?
Chylomicron?
Yeah, the chylomicrons are massive in scale. Now, I understand why they've done this. They wanted to show what's happening up here. And here they wanted to show us the context of those chylomicrons entering into it. But remember, as it goes from micelles to chylomicrons, this structure gets bigger. It doesn't get smaller, right? And again, if you actually spend some time thinking about it, that's going to make sense. Is something this big in scale to the cell pass into the cell this way? Just by simple diffusion? I mean, no, this thing is bigger than the nucleus of the cell. So clearly the scale is off, right? So the scale is off on this here, but as long as you ignore that issue with the scale, it does a great job of describing the process. All right, so a little bit of goofiness with our artist here, but other than that, that is our third pathway, and I don't remember if I saved it or not, so let's go back just to make sure. Questions on that? All right. What is our last pathway? Because I did say 4. We're really 3.5. What's the other major thing that we need to absorb? Well, actually, before we do this, any more questions on the lipid stuff before we move forward? It is the trickier one, so I'll double check one more time. Any more questions on this before we move forward? All right, excellent. So what's the last thing we need to worry about the absorption of?
Water.
Yeah, notice we haven't talked about the 4th macromolecule, nucleic acids, but like we talked about before, all of the breakdown and absorption of that occurs in the small intestine, right? Chemically, it's broken down just in the small intestine. Mechanically, just the same as the carbohydrates, right? No specialized version of that. And it's going to take the blood pathway. So it's a little redundant and a little outside of the point. But you are right. The other major component that we need to absorb is water. Remember, the lumen of our digestive system is outside of the body. We have talked about how we are making a liter of saliva and 2 liters of gastric juice and a liter of bile and a liter of 2 liters of pancreatic juice and a liter of intestinal juice. We're making all of these juices, which are mostly water, and releasing them from our body. That's 7 liters of fluid leaving our body during the course of a day. And can we afford to lose 7 liters of fluid in a 24-hour period of time? It's not a trick question.
No.
Can I put a tap in you like a tree and drain out your sap and take 7 liters from you and you'd be okay tomorrow? No, of course not, right? So the vast majority of what enters our lumen, of the 9.3 liters of fluid that enter our lumen, very small amount of it is actually from the food and the drink we ingest. We ingest about 2.3 liters of fluid during the course of the day, and that's from either our food or from our drink. But the rest is entering it from our own glands. Our own glands are releasing it. So not surprisingly, a massive amount of that needs to be reabsorbed. Now, I think one of the errors that people make is the primary organ where the reabsorption of water takes place is the small intestine. You randomly grab somebody on the street and ask them, hey, where is water reabsorbed in our body? Everybody's going to say large intestine. The reason for that is in our large intestine, as we'll learn about in just a second, or really at this point, we could just call it absorption. Is that's the primary thing our large intestine does. It doesn't do a whole lot of anything else. So reabsorbing water at the right rate so that our feces isn't watery like diarrhea or too dried out, which would happen, which would lead to constipation. So those are processes that we're comfortable and familiar with. So having and absorbing the right amount of water in your large intestine is important for a good function of our digestive system. But the vast, vast majority, over 8 liters, like I said, all of our primary resorption occurs in our small intestine. So our small intestine is the main site of all absorption, not just nutrients, but also of water as well. Excellent. This primarily happens through osmosis, again, and if we think in terms of pathways, it's going to enter the blood supply and it's going to enter the lymphatic capillaries. So the good news is that absorption takes place primarily in the small intestine with a little bit in the large intestine. It passes both the apical and the basal surface via osmosis. But when describing this, you're going to have to describe both the blood vessel and the lymphatic pathways back to the heart. So that'll be the fun part of that question. All righty. Of that About 9.2 liters is reabsorbed. So only about 100 milliliters of water remains in the feces. And that is the final fourth iteration of our food. We started with food, turned it into a bolus, turned it into a chyme, and now here in our large intestine, we will turn it into feces. And with that, we are done with our small intestine and into our large intestine. Questions on that? All right, let's go ahead and take our last break then. Take a last break, 15 minutes. So it looks like we'll come back at 3:05. And we will restart at that point, and I'll start the recording. All righty, I will see you guys in 15 minutes. All right, there we go. So in the small intestine, we've broken down our macromolecules, done most of the absorption, and then the material that is left enters into the large intestine. And again, large in diameter, not large in length. While being a relatively small organ, right? We talked about how the small intestine was 20 feet long, and it only has 3 parts. 3 regions. We have 3 regions here in the large intestine as well, but it's also divided into kind of a bunch of collection of parts. So let's start easy with the regions. With the regions, basically what we have are 2 pouch-like structures. The first pouch-like structure is what is known as the cecum. It is the pouch that collects the chyme as it exits the ileum of the small intestine into the large intestine. We also have a pouch-like structure at the end, and that is the rectum. And in between, we have what is known as the colon. However, as you can also see, there's a lot of specialization for this colon. We have the proximal part, which is the ascending colon because it goes up. Then there is a very sharp turn to that colon known as the hepatic flexure because it's right here next to the liver, or because it's the right side of the colon, it's also the right colic flexure. Either of those 2 terms are acceptable. We then have the transverse colon that travels in the transverse direction. Another sharp turn, that is the left colic flexure, or because it sits right next to the spleen, is also the splenic flexure. That leads into the descending colon and then finishes off with the S-shaped sigmoid colon. Notice we've talked about some of these regions before. Our large intestine is this interesting organ that is a combination of both intra and retroperitoneal. Remember, the ascending colon and the descending colon are both retroperitoneal. They are covered with adventitious. They are located attached to the walls of the abdominal pelvic cavity. However, the remaining structures, the cecum, and the rectum is also, the descending colon, the ascending colon, and the rectum are all retroperitoneal organs, whereas the cecum, the transverse colon, and the sigmoid colon are all intraperitoneal organs. And in fact, if you remember, and you can kind of see it from the blood vessels here, these blood vessels here travel to the sigmoid colon via the mesentery that we call the sigmoid mesencolon that holds it in place. And remember, the transverse colon is held in place by the transverse mesocolon. So we have those 2 mesenteries for those 2 intraperitoneal portions of the large intestine. Like most of the rest of the alimentary canal, it has a mucosa, a submucosa, a muscularis, and either an adventitia or a serosa. It has 2 layers of smooth muscle, as we would expect. Does have some specializations to the mucosa. We'll look at it in a minute, but I do want you to notice there is some distinction to the gross anatomy. Again, we talked about the regions and all of these regions, right? Cecum, ascending, flexure, transverse, flexure, descending, sigmoid, and rectum. However, if you look at the colon portion of our large intestine, you can see it is divided up into these little segmented parts or sections. What is known as a hostrum being the singular, or hostra being the plural. We have to look microscopically to see the specializations of the mucosa, but we can also, as looking at the gross anatomy here, see there is a special enlargement of the longitudinal layer of the muscularis externa. We can see these enlarged bands of smooth muscle that run the length of the organ. These enlarged bands are what are known as the tenia coli. We see that here on the illustration. Notice these enlargements put a little extra tension on the large intestine, which is what causes the pooching or the pouching to form those hostra. So these hostra are basically formed by the tension of the tenia coli, which is an enlargement of the longitudinal muscularis layer. Made a little bit of a mess of this, but if I remove some of this, notice we can see where the greater omentum would attach to the transverse colon. Remember, it comes off of the stomach and drapes down over the front and back up and attaches here. But notice there are other collections of adipose that form associated with the large intestine as well. These are what are known as the fatty appendages or appendices, or also known as the epiploic appendages. The full function of these is not fully understood. It may be stored, it's obviously stored adipose, stored energy, but it may play an important role in helping to provide readily available food for the all of the elaborate bacterial flora and fauna that is located inside of the large intestine as well. So again, its full function is not completely understood, but it is a very common structure associated with the large intestine. And while we're talking about structures associated with the large intestine, our cecum has this long worm-like structure, that vermiform appendage, a vermiform appendix, I should say, that comes off, which, as we know, is part of our lymphatic tissue. As we talked about, it contains seed colonies of bacterial flora and fauna in a protected nature, but it is also filled with lymphatic follicles, which, if they become swollen, can congest this, causing the appendix to swell and become enlarged, appendicitis. And if that structure, which is delicate, ruptures, basically it tears an opening in the cecum, which is where the basically almost feces is located before it's processed with the large intestine. And that would spill all that almost feces out into our abdominal pelvic cavity. And do you think filling your abdominal pelvic cavity with almost feces would necessarily be a good thing?
Very bad.
Yeah, it'd be a very, very bad thing. Exactly. So it's one of the reasons why that appendicitis can be so dangerous, potentially be dangerous when it occurs. All right. So those are the gross anatomy components of it. Questions on that? Excuse me. When we look at the mucosa, as we can see here, notice one of the first things that you should do, again, I'm always trying to look to identify quickly and easily what organ I'm looking at. And the very first thing I usually look at is the muscle layer. How many sublayers do I have to the muscularis here?
2.
2. So that's a dead giveaway that I am not looking at the stomach. That's important because notice as I look up here, I don't see big villi extending out, but I do see some gland-like pits that are forming coming down in here like that. That could possibly confuse me into thinking that it's the stomach, but the fact that I just have 2 muscle layers is a dead giveaway that I'm not looking at the stomach. I do have some gland-like structures, but notice also these gland-like structures are just chock filled with these very clear white cells. What do you think these clear white cells are that I'm seeing inside of all of these gland-like structures?
Goblet cells.
Goblet cells. And what do goblet cells make again?
Mucus.
Yeah, they make the mucin that makes the mucus, absolutely. Right? I know in the stomach, they should be having like parietal cells and chief cells and not so many of these goblet cells. So these invaginations of the mucous membrane, similar to what we saw in the small intestine, are the intestinal crypts. So how do I tell the difference between the large intestine and the small intestine? If both have intestinal crypts, no villi. Notice I don't have villi. If I have villi, it would be the small intestine. If it doesn't, it's the large intestine. And really, it can be that simple. 2 muscle layers. Well, so yeah, I start up here. Gland-like stuff. All right. No villi, though, so that tells me it's not the small intestine. 2 muscle layers. That tells me it's not the stomach. So basically, the only thing that I'm left with is the large intestine. Now notice, any idea what these things are in our submucosa?
Are they blood vessels?
Yeah, you can actually see the blood, the red blood cells inside of those blood vessels. Not surprisingly, because again, we want to absorb some materials from here. It's not surprising that there would be large blood vessels in here. However, is every section going to have those blood vessels in them really huge like that? No, not necessarily. So it isn't a dead giveaway the same way, but we have glands or crypt-like structures, no villi, and 2 layers of muscle. And so just that simply, I can tell that this is the large intestine. So if you use those simple cues, it should be able to easily identify which organ you're looking at histologically. Now, as I look at this, can I tell whether this is the ascending colon or the transverse colon or the sigmoid colon? No, of course not, right? We're not going to be able to make that distinction histologically. Could we do that in the small intestine? Can we tell the 3 regions in the small intestine?
Yeah.
Yeah, we were able to do that, but we're not going to be able to do that for the large intestine. So we just know that this is generic colon, right? Or a large intestine would be fine.
But it is possible, right? To tell
Yes. No, not really. No, there's even an anatomist isn't going to be able to look at the histology of the ascending colon and descending colon and be able to tell them apart. Now, cecum versus colon, yes, right? Colon versus rectum, yes. But, and we'll talk a little bit about the colon. We're not going to look at it histologically, but we will look at the some of the anatomy of it, which will help us to see why that would be the case. But no, yeah, no, I don't know anybody who's able to do that. The differences are too subtle to be able to tell the difference. Yeah. So it's not that I'm just not making you do it. It's just I don't think it's possible. All right. That's it for the anatomy. Let's talk about the function of the large intestine. As I mentioned, there is specialized mechanical activity associated with the large intestine. Remember the presence of the stretch of the stomach, which releases gastrin. causes the ileocecal valve. Oh, I should say that. Oops, let's go back. Hold on. Like pretty much every other organ that we've seen, it is bounded by valves at both ends. Notice here inside the cecum, we can see the large valve where the ileum feeds into the cecum. And we talked about that with the small intestine, the ileocecal valve. And then what's the valve at the end of the large intestine? Or maybe I should say valves, plural.
It's the internal and external rectum or
Anal
valves.
sphincters. No, you got the right idea. There's an internal and external anal sphincter, not one, but two sphincters at the distal end, controlling the movement of feces out of the large intestine. All right, so as I mentioned, oops, now I got to write it back again. With the help of gastrin, our gastrin tells our ileocecal valve to open, allowing the old food to exit the small intestine into the large intestine to make room for the new stuff. So we mentioned for a small kid with a small large intestine, that can be enough to stimulate a defecation reflex. So we talked about, made the joke about how a kid sits down, has 3 bites of food, and then has to run to the bathroom. And while we did it jokingly, there are plenty of times where that happens to little kids. But there are other specialized movements as well. Again, if you think about, if we have 3 pouch-like haustra here, we can have haustral contractions where basically you just get a contraction of the smooth muscle in just one portion of the large intestine. Obviously, this helps to mix the chyme that is being processed with the bacteria that is found in the large intestine. It can help to extract water and nutrients and material from it, and it can help to move it into the adjacent haustras. However, since it can equally go into both adjacent haustras, is it playing a big role in moving it from the beginning to the end of the large intestine? No. So notice we said this process is similar to segmentation. It's about enhancing chemical digestion, enhancing mechanical digestion, enhancing absorption, and things along those lines, but it's not really propulsion. We do have propulsion to move things out of our large intestine, and those are what we call mass movements. These are large, powerful contractions that go over the entire length of our large intestine. Again, these are going to be controlled by or related to the number of times you eat, the volume of the food you eat, the composition of the food you eat. But on average, these mass movements occur about 3 or 4 times a day. Again, like I said, what you eat and how much you eat and how often you eat affect that by primarily releasing gastrin that can affect quite frankly, the speed at which your chyme becomes feces, and then the time at the speed at which that feces enters your rectum, which determines how frequently you defecate. Lastly, there is another neural reflex called the gastrocolic reflex, where again, as the stomach stretches, we need to get the food moving through the large intestine, through the small intestine. So a stretch of the stomach through a local reflex basically increases the motility of the large intestine. Again, get rid of the old food, make room for the new food. The large intestine itself doesn't have any real chemical activity component. It only really produces one thing. As we saw from the intestinal crypts, basically the one thing it produces is mucus. Doesn't produce its own enzymes, doesn't have brush border enzymes, right? Doesn't make any kind of special secretion. But it doesn't need to, because the large intestine houses a massive and elaborate bacterial flora and fauna. Like I mentioned before, we've talked about this numerous times, right? Every single person in this class is infected with E. coli, right? You all have E. coli inside of your body. Should we all be rushing to the emergency room as a result of that?
No.
No, because that E. coli is located inside of our large intestine, where it is beneficial to us, or at least not damaging to us. It's when it gets out of the large intestine and to other parts of the body that it causes problems. While the small intestine is the primary site where most of the absorption takes place, as we talked about, there are going to be some materials that will be absorbed. We're going to reabsorb some water, about 1 liter's worth. Some of the bile salts can be reabsorbed here. Other organic materials that weren't present in the small intestine. So these are organic materials that are formed by bacterial activity. Once the bacteria break them down, then we're able to absorb them, and that occurs in the large intestine. And if you remember correctly, most importantly of all, one example of an organic material that is released by bacterial activity is vitamin B12. And remind me again 2 things. Why is vitamin B12 so important?
It helps create erythrocytes.
Yeah, absolutely. And what was it that allows us to absorb this B12 from our large intestine? What is the chemical signal? It's not quite a hormone, but it is a hormone-like protein.
Intrinsic factor?
Intrinsic factor. And which cell made that again?
Was it the parietal cells?
Parietal cells. Excellent, 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 excellent. Absolutely, excellent. And then lastly, of course, the large intestine is responsible for converting that chyme into the feces, storing that feces until we get it to a socially appropriate location, and then the removal of the feces from our body. Again, B12 is not the only component that is formed by that bacteria. Bacteria also help to break down that biological material to release vitamins like vitamin K and vitamin B5. There's some other electrolytes and especially lipid-soluble minerals that need to be broken down this way. However, are they able to break down everything?
No.
No, exactly, which is why you didn't have a big bowl of grass for breakfast this morning, right? And by big bowl of grass, I don't mean firing up a bowl of marijuana. I mean like went out to your backyard and grazed for half an hour. All right, did anybody do that for breakfast this morning? There are organisms that just eat grass, like cows. So why can cows do it, but not us?
We can't break down cellulose.
Right, exactly, right. They've got something like 86 stomachs with all sorts of different bacteria, and they're able to swallow the food down and spit it back up, make it into a cud, chew it back up, swallow it down into another one of their 86 stomachs, right? It takes them a lot longer to break that down. We're not capable of doing that. Now, hopefully, none of you have ever eaten a blade of grass before, but plenty of you have had corn on the cob, right? And if you forgot you ate corn on the cob a couple of days ago, how can you tell a day or two later?
It doesn't digest.
Yeah. Now, again, we see those kernels in our feces, Now, we do have to be careful about that. You don't actually see the whole kernel. The kernel actually has a cellulose shell, and then there's all that corny goodness inside. Your digestive system is quite capable and easily breaks down the stuff on the inside, but that outer cellulose shell isn't broken down, and so it's not uncommon to see the shell of it, the whole kernel, but you see the shell of that corn in your feces because you're not able to break it down fully. Excellent. All right. Bacteria primarily ferment is how they metabolize. What does that mean? What is it? What does it mean to break things down via fermentation?
Anaerobic activity is required.
Say again?
Anaerobic activity is required.
Yeah, exactly. It's done without oxygen. All right, as we know, that means it's not very efficient. It also means it produces a lot of byproducts, right? Undigested carbohydrates are converted into carbon dioxide gas and methane gas that produces our flatus. Undigested proteins get converted into ammonias and indoles and hydrogen sulfide, which gives the odor to that flatus. And remember, as we also talked about, that bilirubin is broken down into the stercobilin and urobilin. Urobilin, remember, we talked about is reabsorbed and colors our urine, but the stercobilin stays in the large intestine and causes the color of our feces. So one of the things that, especially early on, it's not so much as now, but especially in like medieval times and things like that, a lot of people paid a tremendous amount of attention to the color and the consistency of feces, or the volume and aroma of the flatus that is produced, with, you know, as an indicator of the overall health of an individual. And while there certainly are some indicators in there, it's also a bit better indication of what's going on with the bacteria inside of your lumen of your large intestine as well. All right, so again, the flatus isn't your fault, it's the bacteria's fault. So just remind your loved ones of that. All right, questions on that? All right, excellent. Again, depending on the number of times you eat, the volume of the food, the composition of your food, it takes about 12 to 24 hours for the complete chemical and mechanical breakdown of the chyme in our large intestine. And after that time, the chyme officially becomes feces. Feces basically contains water, a fair amount of either bacteria or the bacterial decomposition components, any kind of undigestible or inorganic material. So like we talked about those kernels of the corn, or if you think back to the very beginning, that penny that your 5-year-old swallowed, right? Clearly there's no absorptive pathways for currency, so it's going to stay in the lumen and come out the other end. And not surprisingly, a fair amount of epithelial cells. Remember, as we talked about in our small intestine in particular, every 2 to 4 days, every 3 days, you have a completely new epithelial surface in your small intestine. Even with the thick alkaline mucus in your stomach, those epithelial cells are being broken down. Think about the hot coffee and the sharp tortilla chip and the acidic wine and all those things that are passing through your mouth and esophagus, right? We are constantly sloughing off those epithelial cells. Again, they are things that are released during that fecal component as well, which is why you should never poop on somebody you don't like's front porch, because if they're so inclined, they could do a DNA test on it and trace it back to you. All right, so use your dogs. All right, questions on that? And then, like I said, the stercobilin pigment, or It could be pigment from the food you ingest as well. If you read that Gulp, they'll tell you about a famous story of a young kid who had thousands and thousands of dollars worth of tests done because he was having pink feces. And they were very concerned about him. And I won't spoil the story for that, but if you're so inclined, I would encourage you to read that. All right. That brings us to the end of our alimentary canal. The same way the oral cavity had some specializations, our rectum and anal canal have some specializations to them as well. Let's start here. Notice again, we are going to have a mucosa, a submucosa, a muscularis, right? This is below the abdominal pelvic cavity, so this would be an adventitia. It is not intraperitoneal. But notice some other specializations that we can see as well. Notice, similar to what we saw in the circular folds, similar to what we saw in the rugae of the stomach, notice there are these large folds that involve the mucosa and submucosa in the rectum. The rectum is big pouch-like structure, kind of like the stomach is a big pouch-like structure. And these enlargements are what are known as the rectal valves. What might their function be?
Maybe to make sure the feces doesn't just fall out.
Absolutely. That's actually a great way of describing it. Absolutely. It's not like the rugae in the stomach where we want to expand it to involve a massive amount of feces, right? It's not necessarily like the circular folds in the small intestine where we want to slow the process down. You've absolutely got the right idea. The goal of these rectal valves are essentially to allow the flatus to leave while the feces doesn't. So it helps to separate, to stop the feces from exiting when you are expelling your flatus, right? So that's basically a fancy way of making sure you don't shart. All right. So there you go. Those rectal valves play a very important role in that. I want you to notice a couple other things though as well. Notice there is a big, let me change the color from pink. A big enlargement of the smooth muscle layer here. The circular layer of smooth muscle. And that's, as we mentioned earlier, our internal anal sphincter. As I mentioned, this is smooth muscle. So what does that tell us about how it is controlled?
It's controlled involuntarily.
Excellent. However, notice also in this, again, our artist is a little wonky here, but if we look closely enough at this, we can see that we have a portion of our urogenital diaphragm. And with that urogenital diaphragm, with that urogenital diaphragm, what's the primary muscle that forms our urogenital diaphragm? Anybody remember from 430?
With the levator ani?
Yeah, the levator ani muscle. This helps to form that urogenital diaphragm, that support structure for the organs in the pelvic cavity. It's going to play a role in regulating urine flow. It's going to play a role in regulating defecation. It plays a role in the passage of the baby out of the vaginal canal, right? That passing of the baby out of the vaginal canal can actually weaken this levator ani muscle. So during pregnancy, women are typically encouraged to do special exercises to strengthen their levator ani and that urogenital diaphragm. And what are those called? Kegel exercises, excellent, absolutely. That contraction of that. Again, I'm sure everybody did a couple of them right there. I did a couple of them. Everybody does whenever you mention them. But notice this, and again, it's a little weird the way that they have drawn this, but there is an enlargement of the levator ani muscle that is just a little deep to the internal, and hence its name, is that it forms the external anal sphincter. Being an enlargement of the levator ani muscle, what type of muscle tissue is it?
Is it skeletal muscle?
Absolutely, it is skeletal muscle tissue, which tells us what about our control?
It's voluntary.
So we're going to have voluntary control. So as we mentioned, there are two valves, two sphincters at the distal end of our alimentary canal, the internal and the external anal sphincters. Obviously, one is internal, one is external, but also one is smooth muscle, an enlargement of the circular layer of the muscularis externa, and the other is skeletal muscle, an enlargement of the levator ani muscle. Notice that isn't the only thing that becomes enlarged in this region. There's some enlarged venous structures. Why we need such large venous structures is not fully understood. Maybe to help to facilitate the absorption of water near the distal end to make sure that the appropriate amount takes place. However, these large venous structures are what are known as the hemorrhoidal veins. You may not think about them on a daily basis unless they become irritated. What happens when these becomes inflamed and irritated?
You get a hemorrhoid?
Yeah, that's what hemorrhoids are. Hemorrhoids are the inflamed irritation and enlargement of these hemorrhoidal veins. Too much irritation, not enough fiber, right? Other problems can cause it as well. Notice there's a couple other specializations to the mucosa as well. Notice there are these raised ridges, these folds that form right at the opening of the rectum, known as the anal columns. This helps in the contraction and compacting of the feces as it is leaving. And then notice also, again, even though this is an illustration, we can see that they've emphasized that there is this division point, what is known as the pectinate line, that actually is the exit point of the rectum into the anus. And remember, as we've talked about, the openings are that non-keratinized stratified squamous epithelial tissue, whereas what type of tissue is going to line our rectum?
Simple columnar.
Yeah. So that pectinate line is basically the transition point from the simple columnar of our rectum to the non-keratinized stratified squamous epithelial tissue of our anus. And of course, the anus is the passageway out. All righty. Questions on this anatomy? All right, we did that, we did that, we did that, we did that, we did all of that. Then the last thing we need to talk about then is, again, the same way we started the process with a reflex, the deglutition reflex, we need to finish this process with the defecation reflex. Again, your book does a nice job of describing this, but again, let's kind of simplify things, look at a schematic of this and kind of understand what we are dealing with. As we know, again, we've got that sigmoid colon that is going to feed into the pouch-like rectum. Hold on, this is taking up too much room. We also know that there are two sets of valves, our internal anal sphincters and our external anal sphincters. So let's get some anatomy on here. Sigmoid colon, that's going to be too big. And again, remind me, our internal anal sphincter is what type of tissue?
Smooth muscle.
Excellent. And then our external anal sphincter is
Skeletal muscle.
excellent. All righty. So here is the basic anatomy. As we know, there are mass movements and other peristaltic events. That might be too big. That are going to, so let's actually do it this way. Hold on. Excellent. So we are going to have I'll avoid using brown. We'll use purple. Mass movements and other peristaltic events are going to move feces into our rectum. And of course, this pouch-like structure, as it fills with feces, is going to push on the walls and cause the rectum to stretch. And of course, not surprisingly, our rectum has stretch receptors. And when they stretch, they become more active. Now, this stretch of the rectum is going to trigger two reflexes. One short and one long. So let's talk about the short reflex first. In the short reflex, basically the stretch of the rectum is going to, using the enteric plexus, it is basically going to do three things. It is going to cause an increase in motility of the sigmoid colon. What's that going to do? The sigmoid colon increases its motility. What's going to happen?
Push
More
more feces.
feces.
Yeah, more feces is going to come to the rectum. It is going to start rectal contractions. Right, this will start packing up the feces and starting to try to move the feces out, and it will cause a relaxation of the internal anal sphincter. trying to open up the passageway for the feces to come out. Now, do you feel these contractions when this starts to occur?
Yes.
Yeah, absolutely. Right. This is when you start being aware of the fact that you need to defecate. However, notice with all of this increased activity, more feces coming to the rectum squeezing that feces, and the internal anal sphincter opening. When all this occurs, does feces just explode out of your body at that point?
No.
No. And that is because there is that long reflex. The long reflex, of course, is going to uses our central nervous system. And in particular, this is a sacral spinal cord. So using those splanchnic nerves, we send a signal to the spinal cord, the sacral region of the spinal cord, which then sends a signal back to the rectum and anus. And this long reflex causes the contraction of the external sphincter. So while you feel the need to void, you're aware of the fact that defecation needs to take place, feces does not explode out of your body because the external anal sphincter contracts. Now, luckily, our external anal sphincter relaxes. And of course, this is, I should also say, this is a parasympathetic reflex that uses the sacral spinal cord for that. Causes this to contract. Then you go to a socially appropriate location. When you get to that socially appropriate location, what do?
Voluntarily relax the external anal sphincter.
Now, with that voluntary relaxation of the external anal sphincter, both sphincters are relaxed. And what happens? Not a trick question. Nobody wants to say defecation occurs. The poop comes out. Bingo. Excellent. Which is all fine and all dandy and hopefully makes a whole heck of a lot of sense. Except for one minor issue. What if all of this is happening as you're sitting there in your car driving and you just passed a sign that says the next rest stop is 30 miles? What happens then?
You speed up.
Okay, true. You can speed up, but what happens?
The external sphincter is less and less relaxed.
Okay, possibly, but is that really what happens the first time? If you're sitting here in the middle of lecture talking to me and you suddenly felt the need to void, right? Again, and assuming we were in the classroom and not actually here where you could be pooping for all I know right now, right? Are you going to necessarily go running out of the classroom as soon as you feel that need?
No.
No, because you can choose not to relax the external anal sphincter. And again, in the car, you have that choice. Sure, you can pull over to the side of the road and find some bushes, or you could keep driving for the 30 miles. Yeah, you might speed up a little bit, but it's still going to take you some time to get there. Do you feel the need to void the entire time that you're driving those 30 miles?
Yes.
Really? You feel the contractions, you feel the need to void that
Oh,
entire
feel it.
time? No, this sensation habituates. It goes away. So if you don't void right away, after a few minutes, the contractions stop. The need to void, the internal anal sphincter contracts again, and it will habituate. So you're not feeling the need to void the whole time. However, the rectum is going to continue to fill. And as it continues to fill, right, the pressure becomes greater. And as the pressure becomes greater, stretches again, reaches a critical point, and the contractions become more powerful. And you can try to hold it again, and it eventually will go away again. But if you continue to do this, what eventually can happen? Can you just refuse to defecate forever?
No, you're just gonna
Right.
defecate.
No, the first year that you date a man, I know you're not supposed to defecate at all during that time. I know I didn't the whole first year I dated a man, right? You're not supposed to defecate, right? They're supposed to just think you as this delicate flower, but you can't really hold it for an hour. I mean, for a year. Eventually, right, the contractions can become powerful enough where the force of the contractions can become great enough to force the feces out, whether you want it to or not. Of course, the other is true as well. One of the problems is by the time you get to that rest stop, because it's habituated, now your internal anal sphincter isn't as open, it's closed again, the rectum's not contracting anymore. So now when you finally get to the rest stop and get to the bathroom, defecation may not necessarily be easy, right? So luckily we have ways to assist And how are some of those ways that we can assist the process?
By contracting the abdomen.
Okay, excellent. When we contract the abdomen, what does that do?
It increases the pressure we're putting down.
Yeah, it increases the pressure in the abdominal pelvic cavity. And can help to force the feces out. What else can we do? Well, remember, we can also elevate that levator ani muscle by elevating the levator ani muscle. That can help to open the sphincters mechanically. It's kind of in a similar way to, as we saw, the opening of the upper esophageal sphincter when the larynx elevates, can help to open the sphincters to help to facilitate defecation. And there's one other thing that we can do to help to increase the pressure in the abdominal pelvic cavity. One way we can do that is by exactly lowering the diaphragm. Excellent. The way you do that is basically you close your glottis, the airway that we use to control the movement of air. We can close our glottis and then contract our thoracic cavity. When we contract the thoracic cavity with the glottis closed, instead of the air coming out, it pushes the air down and basically it depresses. So we can use air to depress the diaphragm. And that will also increase the pressure in the abdominal pelvic cavity. Can help to that as well. Of course, they have a fancy name for this. This is known as the Valsalva maneuver, where again, that bearing down, if you think of that bearing down, when you're bearing down, you contract your abdomen, you force the air down, contracting your thoracic cavity, try to increase that pressure to help in the removal of the defecation. Notice though, one aside to this, notice both this contraction of the abdominal pelvic cavity and the Valsalva maneuver play a role in increasing the pressure of the abdominal pelvic cavity. If pressure becomes too great in the abdominal pelvic cavity, right, we know that that pressure can kind of try to force its way out in other areas. And what do we call that when we get a weakening of the muscles and the expulsion of fluid or even organs in the abdominal pelvic cavity from too much pressure? Come on, I know you know. Most people think of these things occurring when you're doing something, you know, big and strong and womanly, like, you know, moving a sofa or lifting a car.
Hernia?
A hernia, absolutely, right? We can get an umbilical hernia. We can get an inguinal hernia for males, much more common in males than in females. Some studies believe that as many as 30% of hernias occur via defecation. It is a hard number to track down because, surprisingly, some people are unwilling to admit that they got a hernia from pooping too hard, right? So again, since these things are things that are self-reported, we don't have cameras in our bathrooms yet. Give time, I'm sure we will eventually, but at this point, you're not being monitored while you defecate. And so again, self-reporting is the way that we know the causes of hernias. And since some people surprisingly are reluctant to admit that their hernia occurred when they were bearing down during defecation, but some studies show that it may be as high as 30% of umbilical hernias occur via defecation. So don't bear down too hard. All right, questions on that? All right, we started with cheeseburgers, we end with pooping. We have done it. We have made our way through the entire alimentary canal. Questions on any of that? All right, well, that is all of the new information that I have for you. If you look at the, I mean, again, there are still some lecture slides, but if you look at the lecture slides that are there, again, we just talked about the defecation reflex. We have that there. We talked about that. Talked about the assistance, the things that we talked about for that. And then there's this nice summary table. So again, nothing we didn't already cover that's on there. That is the digestive system. And that is all that you're responsible for on this next exam. Absolutely, this video will be up before the exam. I'll try to get it up tonight. If it doesn't, it's all about how long. The two things that delay it is how long it takes to compile it on my computer, because that's where I record it. I know you can do it in the cloud, but I had a lot of problems with that originally. So first thing I have to do is compile it here. Then when I upload it to YouTube, it takes time for it to process. But usually it's the first day. So if not by tonight, then tomorrow morning, definitely I will have it posted. And I will definitely, especially with the test on Thursday, I will try to get this done today. All right. Any other questions?
I have a question on some like older material. It's still digestive system, just
absolutely.
Could you explain what the lobules of the salivary gland are, or where they're located?
So this is actually a great question, because it actually works the same with our liver as well. Basically, a gland, and again, the salivary gland is a good example of this. You have this big gland, all right? This gland typically isn't just one uniform structure. Typically what happens is we have connective tissues that divide it into individual compartments. And these individual compartments are the lobules. They're primarily significant from a structural standpoint, because typically one lobule will have one main duct that feeds into it and one main blood supply that goes into it. So one main artery that goes into it, one main artery that goes out. So typically it's just, we know in, for instance, that salivary gland, we're going to have a bunch of little ducts and we're going to have a bunch of some type of secretory structure, whether it's tubes or whether it's a cini. But that's not just continuous throughout the entire gland. There is going to be compartments, individual compartments divided by connective tissue. So this connective tissue, basically this dense irregular connective tissue, subdivides the glands into individual lobules. We kind of just saw that. I'm going to sheet here and use the same thing. Oops. Cut it in half. So instead of the salivary gland, if we were thinking in terms of the liver, as we mentioned, the liver is made up of a bunch of hexagons. I know my hexagons aren't the best. And we know, and again, I'll just put a single circle there. There are the portal triads at the center of these, like places like that. Well, these portal triads are basically located in the connective tissue septums that divide the lobules into individual compartments. So there are a bunch of connective tissues that basically subdivide this into all of the individual lobules. So a lobule is basically just a functional unit of a gland or of an organ, typically divided by connective tissue septums.
Awesome. Thank you.
Yep, it's a great question. Any others? The only stupid question is the question not asked. Okay, so two big differences between this. So in our saliva, there are two main enzymes that are produced from two different locations. As the name indicates, lingual lipase comes primarily from glands in the tongue, whereas salivary amylase comes from those three glands that make our saliva, the parotid, the submandibular, and sublingual. So those are the, they're enzymes that are coming from different locations. Actually, I guess there's three. There's two other differences as well. As the name indicates, they break down different things. Amylases are responsible. Remember, ACE is just the suffix that means something is an enzyme. It breaks something down. So a lipase breaks down lipids. That one's easy. Amylase is a little trickier because ama doesn't sound like any of the macromolecules that we've learned, but you just have to know that amylase breaks down carbohydrates. So salivary amylase and amylase breaks down carbohydrates. Lipases break down lipids. The third difference between the two is that our salivary amylase is released in an active form. So as we talked about, if you take a piece of bread and put it on your tongue, as the saliva mixes with it, you will start to taste sweetness because the salivary amylase will start to break it down into simple sugars, and you'll be able to taste those simple sugars as it breaks it down. The lingual lipase is inactive. It's in an inactive form. So you could put a chunk of butter on your tongue and it could sit there for days, and the lingual lipase isn't going to be able to do anything to it. However, when you swallow it down into your stomach, the pH change activates the lipase so that it is able to break it down. So those are the three big differences between those. One's produced active, one's produced inactive, one's made by the salivary glands, one's made by the tongue, and one breaks down lipids and one breaks down carbohydrates. Not a stupid question at all. All right, excellent. Any other questions?
Professor?
Yeah.
For the papillae on the tongue, are the filiforms the one that don't contain any taste buds?
So yes, filiform are the pointy ones that don't have and never had taste buds. The foliate, remember, on the lateral aspects of the tongue, start in infancy with taste buds, but those go away. But those you're not responsible for histologically. For those, you'd only be responsible for the gross anatomy of them.
Okay. Okay, thank you.
Yep. Great questions. Any more? Again, remember, if you don't know what questions you have yet, I remind you, I do have office hours tomorrow. I've got class from 8 to noon 30. So basically from noon 30 to 1:30, I have my office hours. And I also have office hours the hour before our test. So that's always, it's kind of last second, but if you have something that's really confusing you, that's always a good time to ask it. And then of course, I'm always good at responding to emails. I'm always good at responding to emails, but especially around test time, I try to get a little, make sure I'm good about that. So if you don't have any other questions right now, it's okay to save those or to think about them or come up with something and let me know before the exam. All right.
Professor.
Yes.
So I know that when we're talking histologically, I know that you always mention we should be looking at the villi, look at that submucosa, look at the muscle, the muscularis externa. But I'm still having some trouble differentiating a couple times. So like, could you explain the difference to histologically between like the colon and like the small intestine?
Well, let's cheat a little bit. And by cheating a little bit, I mean, again, it's never good to ask questions that you don't actually know the answers to, but let's do that. Because imagine there's something here. I don't know how good it is, so there's always that. But on this Cosumnes River site, we have digestive system and we have histology. So let's see what their histology is. And from looking at these things, see if we can figure out blindly without knowing. I don't know either the order or what's here. Let's see if we can figure it out. What do you think this is? Anyone hazard a guess as to what they think this is?
The tongue?
Yeah, it's the tongue. How did you know that?
It doesn't really have any layers, but it has like a top rough surface.
And a thick. Notice we can see many cell layers. Even at a low magnification, we can see there are many layers of cells. And even these top ones have nuclei, so we can kind of see that. Deep, even though we didn't talk about it a lot, remember we talked about how there's all this skeletal muscle at all sorts of different orientations, those intrinsic and extrinsic muscles. So notice these are muscles cut at cross section. Here we have some oblique sections through the muscles. Here we have some longitudinal muscle fibers. Now, if I put a star right here, can I tell that this is whether this is intrinsic or extrinsic muscle? No, of course not. But we know it's that skeletal muscle in here. And notice we can even see some of those glands that are making the mucus and would be making the lingual lipase. Lastly, notice we have this nice big large papillae, right? So we can see that papillae with that nice big huge large valate papillae, big huge valate papillae that we can see in here. So notice we can see some of those simple things. And again, novel looking at it, there's that circumvallate papillae, again, nice big huge papillae. However, notice now at a closer magnification, we can more clearly see multiple layers of cells with nuclei at the top. But the other thing that we can see here is notice there are these clear circle things here on the side. Any idea what those things might be?
Taste buds.
Yeah, those are the taste buds, absolutely. So those are some things that are pretty nice and obvious on this. Again, that's deep in the tongue. That's not something you're responsible for, so I don't really care. Oh, what this is supposed to be. Oh, okay, cool. Excellent. So notice we have a duct. We have a second duct. And we have all of these very clear kind of adipose looking cells around it here. So this is one of our salivary glands. Which salivary gland would this one be?
Sublingual.
Sublingual, excellent. What substance is this secretory structure making?
Mucin.
Mucin. What is the shape of this secretory structure?
Tubular.
Tubular, excellent. So, big three questions. What is the structural classification of this gland?
Compound tubular.
Excellent. What is the functional classification of this gland?
Aerocrine.
Excellent. What is the substance it produces?
Mucin.
Mucin, right, absolutely. Which is a component of what?
Mucus.
True, well, okay, it's going to become mucus and become our saliva ultimately, right? But yeah, mucin is what I was looking for there. Excellent. So then I'm guessing, excellent. So what do we have here?
Pancreas.
Excellent. Notice we see the acini all around there and that pancreatic islet. That's one of the things that was a big dead giveaway. So again, remember, this is our mixed gland. With this mixed gland, we have our endocrine component, that pancreatic islet or islet of Langerhans. Notice there's another one up here. And all of the small clusters that are the pancreatic acini that are producing the bicarbonate and all those enzymes and all those things that we talked about. Excellent. Right. Notice again here, they're showing us one of those pancreatic islets up close. Remember, we've got alpha, beta, delta, and gamma cells in here, but you're not going to be responsible for that, so we don't care about that. But this we care about. What are we looking at here?
The liver.
Liver, excellent. We can clearly see the lobule. And notice, as you were mentioning before, we can see the connective tissue septums that are dividing them out. At this magnification, we can clearly see the central vein. We know the portal triads in these areas, but maybe with one exception, we really can't tell what they are. I would not ask it at this magnification, but based on what you know, what do you think this particular structure right there might be?
The artery.
That big in this magnification?
Is it the triad?
It's one of the three structures of the triad. And which one's going to have the largest lumen?
The vein.
Yeah, but
The hepatic portal vein?
hepatic portal vein. Excellent. Absolutely. And again, not a fair question that I would ask on the exam. We're just kind of working our way through it here. Hopefully they will have, okay, there we see the central vein, the hepatocytes, the sinusoid capillaries really nicely. Ah, here we go. So notice, what is this structure right here?
Hepatic portal vein.
Hepatic portal vein. What is this structure right here?
Bile duct.
And what is this structure right here?
Hepatic artery.
There you go. Smooth muscle, simple squamous. Smooth muscle, no smooth, very little smooth muscle, simple squamous, and the largest lumen. And then those cuboidal cells. I know the histology of this can be scary, but these things are so obviously different from each other. Hopefully that portal triad should be something that's super simple to be able to distinguish. Excellent. Now this one's not a fair question, so that's why I'll show you the picture of this here. Notice the things that we were talking about before. Notice we are seeing that there's some big invaginations of our mucosa, right? So as we mentioned, that could mean it's either the small intestine, the large intestine, or the stomach. So remember, the place we were supposed to look next is going to be the muscularis externa. However, can we see all the layers or potential layers of the muscularis externa?
No.
No, so that makes this a trickier question. Now, we don't see villi, so which ones can we knock off right off the bat?
Small intestine.
Small intestine. So now we're down to a 50-50 shot of stomach or large intestine. This is indeed the stomach, and I will show you how to do this, but I would hope that I would have an obvious enough example where you would be able to see that. Remember, with the stomach, we have those mucus cells that form the pit. And then down in the gland, we have the parietal cells and the chief cells and the enteroendocrine cells. And see how dark all these stains on these cells are? In all of this down here, do you see any goblet cells at all?
No.
No. Remember, our intestinal crypts are going to contain a large amount of goblet cells. And this clearly has no goblet cells. So this is clearly a stomach gland, a gastric gland, making that gastric juice. Although, like I said, obviously without the muscularis layer, it does make this question a little bit more tricky. We were able to tease it out. And even without being sophisticated, without the villi, we were able to clearly get it down to a 50-50 of what it might be. But this one is, I will admit, is a little tricky, right? Again, they're showing us those deep glands. And remember, I'm not going to hold you responsible for those glands in there. And again, they're not showing us all of the muscularis externa, so it's hard to tell if this is the stomach, right? Again, they're showing us the difference here between the pits. Remember, the pits just have mucus cells, lots of goblet cells that we're seeing on here. And then down in the gastric gland, the cells are very, very different. So that is one of the ways that you can tell the stomach from the small intestine, the difference of the consistency, the large intestine. The large intestine, it's going to be goblet cells the whole way. There are clearly different types of cells down here than there are up here at the top. So there's a difference between our gastric pit and our gastric gland. You're not going to see that difference in the large intestine. All right. Excellent. What are we looking at here?
Small intestine.
Okay, but be more specific. Which region of the small intestine?
The duodenum.
Right, excellent. Notice in this case, we can definitely see 2 layers of smooth muscle. We don't know whether or not there's a third. So how are we so certain that this was the small intestine?
Villi.
Villi. So as soon as you see villi, you know small intestine. And when you know small intestine, where do you go next?
Submucosa.
Bingo. Because that tells you which specific region. And what in here told you that this was the duodenum?
With the duodenal cells.
Duodenal glands or the Brunner's glands. Absolutely. There's those big, again, remember they're producing bicarbonate and mucus to help to neutralize the chyme. Excellent. Bingo. Right. Notice here we're seeing a nice look at those intestinal crypts. Notice the intestinal crypts do kind of look like the gastric glands, but notice it's goblet cells all the way down. So all these, there's no difference between the top and the bottom. And notice we have the advantage of still seeing the villi, and we can still see a duodenal cell. So duodenal gland. So we are still in the duodenum. Excellent. What's this?
Would this be the jejunum?
Yep. Notice again, we can't truly see how many layers of smooth muscle we have here, but we clearly see villi. Notice again, we're seeing those crypts, the intestinal crypts, right? But as soon as we see villi, where do we look?
Submucosa.
And what do you see in the submucosa?
Not much.
Not much, which tells you indeed that this is the jejunum. Again, remember, the jejunum does have the longest villi, and this is kind of obvious on this picture, but remember, that can be somewhat subjective, and when you're not seeing the pictures back to back and back, it can be hard to tell, oh, is that a long one, or is that a short one, or a medium one? You know, what am I looking at there? There's no ambiguity in the submucosa. This one's got nothing in the submucosa, so this is clearly the jejunum. Again, showing us the intestinal crypts, showing us the villi. We don't care about that. And what do we have here? Identify the region of the organ.
That's the ileum.
Yeah. Notice we still see villi, much, much shorter. We still see intestinal crypts, not as many. But what's the big dead giveaway inside of our ileum? Those Peyer's patches, absolutely. Peyer's patches in the submucosa. Super simple, easy, easy. So what is this?
The large intestine.
Exactly. This is the colon. Notice, again, first easiest thing is we don't see villi. So we know it's not the small intestine. We do see these gland-like or crypt-like structures. So that means it's either the large intestine or the stomach. We can't see all the muscularis, so we don't know there. So remember, we looked at the consistency of the cells. Are there clearly different cells at the top than there are at the bottom?
No.
No, they're all pretty uniform. And we clearly see some goblet cells down here near the bottom. And so again, that can be a bit of a giveaway that sure enough, we're in the colon. Now, here's the one warning I will give you. Remember, in the stomach, and they didn't show it to us here, so I didn't mention it at the time, we do have to tell the difference between the pyloric region and like the fundus or the body. The ones we were looking at, this is the fundus or the body of the stomach. And there are two ways that we'll be able to tell that. In the pyloric region, there are going to be more mucus glands the whole way. It pretty much only produces mucus. However, the dead giveaway that you are in the pyloric region is it is going to have a massive smooth muscles. The 3 layers of smooth muscles are going to be huge because remember that pyloric region is forming some really, really powerful contractions. I make a point of emphasizing this because you're not going to see villi. You're going to see some mucus glands that go all the way down. And so your first thought might be, this must be the large intestine. But you will see 3 very distinct layers to the mucosa externa. And so as a result, it'll be a dead giveaway you're in the pyloric region. So that's the only thing that could be a little tricky. The pyloric region of the stomach can look a little bit like this, but the mucosa is going to be way bigger. So these are going to be way deeper mucus glands, and it'll have a massive muscular layer that'll be really, really obvious. And I'm guessing, well, there's those intestinal crypts again. There you go. Theirs was pretty good. Almost everything we wanted to see they had there. So that was pretty good. So that's exactly the kind of things you could expect to see on the exam. And that's exactly, with one organ system, probably not as much as the first one, because obviously that one was cardiovascular, had much. I don't know. My guess would be probably somewhere between 60 and 70, maybe. I haven't finalized this one. So there's a lot of histology and a lot of anatomy. Each organ is so unique. So my guess is probably somewhere between somewhere around 60 to 70 would be my guess, somewhere in that range. I think that's reasonable. All righty, excellent. And look at that. We filled up the rest of the extra time we had. These were great questions. I think we got time for one more. Did anybody got one more question, or are we going to call it a day? We were recording good. So again, I appreciate that we did that histology a little quick, but at least now you have it on the recording if you need to go back and look at it. We went slower through each individual organ, but it was kind of fun to do it all at once like that as well. So hopefully that was helpful. Like I said, if you have any more questions, please feel free to email me or come to my office hours either tomorrow or Thursday before class. Otherwise, I guess I will see you in a week. So study hard, do well on your exams, do your pre-labs, and I will see you when we dive into the respiratory system. Take care, guys. Have a good day.
Thank you.